Tonight, we will examine the complete science of coral reefs. We will move slowly through their biology, geology, chemistry, ecology, evolution, distribution, and environmental importance. You may listen for as long as you wish, and you may fall asleep at any point. A coral reef is a biological structure built mainly from calcium carbonate. Most of this material is produced by reef-building corals, although many other organisms also contribute. Coralline algae, mollusks, foraminifera, bryozoans, tube-building worms, and microorganisms can all add mineral material to a reef. Coral reefs occur mainly in shallow tropical and subtropical seas. They cover only a small fraction of the ocean floor. Most estimates place their total area at less than 1% of the seafloor. Despite this limited area, reefs support a very large share of marine biodiversity. A reef is not a single organism. It is a community, a geological structure, and a chemical system at the same time. Living organisms build the reef. Physical forces break it down. Chemical reactions dissolve and rebuild its minerals. Sediments accumulate in gaps. New generations grow on the remains of older ones. The word coral describes animals belonging to the phylum Cnidaria. This group also includes jellyfish, sea anemones, and hydroids. Corals are placed mainly within the class Anthozoa. The name Anthozoa means flower animals. Members of this class usually remain attached to a surface during the adult stage. The basic coral animal is called a polyp. A polyp has a soft cylindrical body. Its lower surface is attached to a substrate. Its upper surface contains a mouth, surrounded by tentacles. The mouth leads into a central digestive cavity called the gastrovascular cavity. Corals do not have a separate stomach, intestine, and circulatory system in the way vertebrates do. Digestion begins inside the gastrovascular cavity. Nutrients then move through tissues and, in colonial species, through connections between neighboring polyps. The tentacles contain specialized cells called cnidocytes. Each cnidocyte holds a structure known as a nematocyst. A nematocyst is a microscopic capsule containing a coiled thread. When triggered, the thread rapidly discharges. It may pierce, entangle, or adhere to a small prey organism. Nematocysts are among the fastest cellular structures in the animal kingdom. Their discharge is driven by pressure and chemical gradients inside the capsule. The process can occur in a fraction of a millisecond. Most reef-building corals belong to the order Scleractinia. They are often called stony corals or hard corals. Their polyps secrete external skeletons made mainly of aragonite. Aragonite is one crystalline form of calcium carbonate. Calcium carbonate can occur in several mineral forms. The two most important biological forms are calcite and aragonite. Reef-building corals usually produce aragonite. Many algae and other organisms produce calcite or high magnesium calcite. A coral polyp sits within a skeletal cup called a corallite. The corallite has vertical partitions known as septa. These partitions provide structural support and increase the surface area where living tissue contacts the skeleton. As a coral colony grows, each polyp adds new skeletal material beneath and around itself. The living tissue usually occupies only a thin layer near the outer surface. Most of the colony consists of older skeleton. Colonial corals are made of many genetically identical polyps. These polyps are produced through asexual reproduction. They remain physically connected by living tissue. Depending on the species, nutrients and chemical signals can move between them. Different coral species form different colony shapes. Some produce massive rounded forms. Some form branching structures. Others grow as plates, columns, ridges, thin crusts, or solitary cups. Massive corals often grow slowly, but can survive strong waves and physical damage. Branching corals may grow more rapidly and provide complex habitat, but their narrow branches can break during storms. Plate-like corals increase their horizontal surface area. This can help them collect light in deeper or shaded water. Thin plates are less resistant to heavy wave action, so they are more common in protected locations or below the shallowest reef zones. Coral form is influenced by genetics and environment. the same species may grow differently under different light levels, water movement, sediment conditions, and competition. This variation in form is called phenotypic plasticity. Many reef-building corals live in symbiosis with microscopic algae. These algae belong mainly to the family Symbiodiniaceae. They were once commonly grouped under the informal name Zooxanthellae. Symbiodiniaceae are dinoflagellates. Dinoflagellates are single-celled eukaryotic organisms. Some are free-living, some are parasitic. Some form symbioses with animals. The symbiotic algae live inside cells in the coral's gastrodermal tissue. Each algal cell is surrounded by a membrane structure formed by the coral host. The coral regulates the chemical conditions around the algae. The algae carry out photosynthesis. They use light energy to convert carbon dioxide and water into organic compounds. Oxygen is released as a byproduct. A significant portion of the carbon fixed by the algae is transferred to the coral. The transferred material may include sugars, glycerol, amino acids, and other small organic molecules. These compounds provide energy for coral metabolism. The coral provides the algae with access to light, dissolved nutrients, carbon dioxide, and protection inside its tissues. Nitrogen and phosphorus released through coral metabolism can be reused by the algae. This symbiosis allows reef-building corals to survive in nutrient-poor tropical waters. Tropical oceans often appear clear because they contain low concentrations of plankton and dissolved nutrients. Coral reefs maintain high productivity by recycling materials very efficiently. The coral-algal relationship is not identical in every species. Different coral species host different symbiont types. A single coral colony may contain several symbiontes. The dominant type can change over time. Some symbionts support rapid coral growth under stable conditions. Others are more tolerant of heat or strong light. More heat-tolerant symbionts may transfer less energy to the host under normal conditions. This creates a possible trade-off between growth and stress resistance. Corals are not completely dependent on photosynthesis. Most species can capture plankton and suspended organic particles. They may also absorb dissolved organic compounds from seawater. At night, many coral polyps extend their tentacles. Zooplankton moving through the water can be captured by nematocysts and move toward the mouth. Mucus on the coral surface can also trap small particles. Heterotrophic feeding supplies nitrogen, phosphorus, and other nutrients that may be limited in clear tropical water. It can become especially important when photosynthesis is reduced by cloudy conditions, deep water, bleaching, or sediment. Corals produce large quantities of mucus. This mucus protects tissue, removes sediment, traps food, and supports microbial communities. Some of it enters the surrounding reef food web. The mucus may dissolve into the water or form suspended particles. Microorganisms consume it. Small animals consume those microorganisms. In this way, coral mucus transfers energy from coral colonies into surrounding reef habitats. A coral colony contains more than coral animal cells and algal symbionts. It also contains bacteria, archaea, fungi, viruses, and other microorganisms. Together, the coral animal, its symbionts, and associated microbes are sometimes described as a coral holobiont. The holobiont concept emphasizes that coral health depends on interactions among many organisms. Some microbes help cycle nitrogen or sulfur. Some may produce antimicrobial compounds. Others can become harmful under stressful conditions. Coral-associated bacterial communities vary among species and locations. They can change with temperature, disease, pollution, depth, and season. Researchers are still determining which microbial changes are causes of stress and which are consequences. Nitrogen is especially important in coral symbiosis. Both the coral and its algae need nitrogen to make proteins and nucleic acids. However, too much dissolved nitrogen can disturb the balance between host and symbiont. Under normal nutrient-poor conditions, algal growth is limited. The algae transfer a substantial amount of fixed carbon to the coral. When nitrogen becomes unusually abundant, algal cells may retain more carbon for their own growth. Excess nutrients can therefore reduce the efficiency of carbon transfer. They can also increase algal growth on the reef surface. This may place corals at a disadvantage. Phosphorus limitation can also affect coral metabolism. An imbalance between nitrogen and phosphorus may damage cell membranes and increase sensitivity to heat. The effects depend on nutrient form, concentration, duration, and local conditions. Reef-building corals produce skeletons through calcification. Calcification occurs in a narrow space between the living tissue and the existing skeleton. This region is called the calcifying fluid or extracellular calcifying medium. The coral actively modifies the chemistry of this fluid. It transports ions, removes protons, and raises pH relative to the surrounding seawater. These changes increase the concentration of carbonate ions needed for aragonite formation. Calcium ions and carbonate ions combine to form calcium carbonate. In simplified form, the reaction can be written as calcium plus carbonate becoming calcium carbonate. The actual biological process involves enzymes, organic molecules, ion channels, and cellular transport systems. An enzyme called carbonic anhydrase helps convert carbon dioxide and water into bicarbonate and hydrogen ions. Bicarbonate can then participate in photosynthesis and calcification. Coral tissue also contains organic matrix proteins. These molecules influence where mineral crystals begin and how they grow. The skeleton is therefore not simply inorganic material deposited at random. Photosynthesis can promote calcification in several ways. It removes carbon dioxide, raises local pH, supplies energy, and alters ion transport. For this reason, many reef-building corals calcify faster in light than in darkness. Calcification continues at night, but often at a reduced rate. Nighttime skeletal growth may have a different crystal structure from daytime growth. Coral skeletons record environmental information in their density bands and chemical composition. Massive corals can produce annual bands similar in principle to tree rings. Researchers use these bands to estimate age and past growth rates. Ratios of chemical elements in the skeleton can reflect seawater temperature and chemistry. Oxygen isotopes, strontium to calcium ratios, boron isotopes, and other signals are used in paleoclimate studies. These records are not always simple. Biological processes can modify the chemical signal. Changes in growth rate, light, reproduction, or tissue thickness may affect skeletal composition. Coral colonies can live for decades or centuries. Some massive colonies may persist for several hundred years. A colony can survive even when parts of it die, because living tissue may remain elsewhere on the skeleton. Individual coral polyps are usually much shorter-lived than the entire colony. They are replaced as the colony grows, buds, and repairs itself. The genetic individual can therefore be much older than any single polyp. Corals reproduce both sexually and asexually. Asexual reproduction expands existing colonies. Sexual reproduction produces genetically distinct offspring. Budding is a common form of asexual reproduction. A new polyp develops from an existing polyp. In intracollicular budding, the new polyp forms inside the wall of the parent corallite. In extracollicular budding, it develops outside the existing corallite wall. Fragmentation is another form of asexual reproduction. A piece of coral breaks away and survives independently. If it lands in a suitable location, it may attach and form a new colony. Fragmentation is especially important in some branching corals. Storms can scatter fragments over a wide area. Many fragments die, but some establish new colonies. Sexual reproduction occurs in several ways. Some corals release eggs and sperm into the water. Others fertilize eggs internally and release developed larvae. Corals that release eggs and sperm together are called broadcast spawners. Large numbers of colonies may spawn during the same night. This synchronization increases the chance of fertilization. Spawning time can be controlled by several environmental signals. These include water temperature, day length, tides, sunset time, and lunar phase. On many reefs, mass spawning occurs several nights after a full moon during a particular season. The timing differs among regions and species. Some colonies are hermaphroditic and produce both eggs and sperm. Others have separate male and female colonies. Some species release bundles containing both gamete types. After release, egg and sperm bundles float toward the surface. The bundles separate. Fertilization occurs when sperm from one colony encounters eggs from another compatible colony. Fertilized eggs divide and develop into planula larvae. A planula is small, usually elongated or oval, and covered with cilia. The cilia allow it to swim. Planula larvae may remain in the water for hours, days, or weeks. Their duration in the plankton depends on species and environmental conditions. Some larvae settle close to the parent reef. Others can travel many kilometers. Ocean currents influence their paths, but larval behavior also matters. Larvae can respond to light, gravity, pressure, sound, chemical signals, and surface texture. Many prefer crustose coralline algae or surfaces with particular microbial films. Crustose coralline algae are red algae that deposit calcium carbonate in their cell walls. They form hard pink, red, purple, or gray crusts over reef surfaces. Certain chemicals released by coralline algae and associated bacteria can trigger coral settlement. After settling, the planula changes into a primary polyp. The primary polyp begins secreting a skeleton. It then produces new polyps through budding. A colony gradually develops. The early life stage is highly vulnerable. Newly settled corals can be covered by sediment, eaten by grazers, overgrown by algae, or killed by temperature stress. Only a small fraction of coral larvae survive to become adult colonies. High mortality is normal in marine organisms that produce large numbers of small offspring. Some coral species brood their larvae internally. Fertilization occurs inside the parent polyp. The released larvae are often more developed and may settle quickly. Brooded larvae may already contain algal symbiont. In other species, larvae begin without symbionts and acquire them from the environment after settlement. Transmission of symbionts from parent to offspring is called vertical transmission. Acquisition from the surrounding environment is called horizontal transmission. Horizontal transmission allows juvenile corals to associate with symbionts available in the local habitat. Vertical transmission preserves an established parent-symbiote combination. Coral recruitment is the addition of young colonies to a reef population. Recruitment depends on adult reproduction, fertilization, larval survival, transport, settlement, and post-settlement survival. A reef may receive larvae produced locally. It may also receive larvae from distant reefs. The exchange of larvae among reefs creates population connectivity. Connectivity is important for recovery after disturbance. A damaged reef with few surviving adults may depend on larvae arriving from healthier areas. However, strong connectivity can also spread diseases or reduce local genetic differences. The effects depend on current patterns in species biology. Coral populations contain genetic variation. Some colonies are naturally more tolerant of heat, sediment, disease, or low pH. This variation provides material for natural selection. Adaptation occurs when heritable traits that improve survival or reproduction become more common across generations. Coral generation times can be long, but some species mature within a few years. Acclimatization is different from adaptation. Acclimatization occurs within the lifetime of an individual. It may involve changes in gene expression, physiology, tissue thickness, symbionts, or microbial communities. A coral exposed to moderately warm conditions may sometimes tolerate later heat better. This effect is called thermal acclimatization or environmental memory. The strength and duration of this effect vary. Prior exposure can provide protection in some cases. In other cases, repeated stress weakens the colony. The earliest coral-like reef builders appeared long before modern corals. Reef construction has occurred at many times in Earth's history, using different groups of organisms. During the Paleozoic era, reefs were built by organisms such as archaeocyaths, stromatoporoids, tabulate corals, and rugose corals. Rugose and tabulate corals disappeared during the end Permian mass extinction about 252 million years ago. Modern scleractinian corals appeared during the Triassic period, more than 200 million years ago. Their early history followed the largest mass extinction known in the fossil record. The relationship between early scleractinian corals and photosynthetic symbionts is still studied. Some ancient species likely had symbionts, while others may have lived without them. Reef systems expanded and contracted repeatedly through geological time. Sea level, temperature, ocean chemistry, nutrient conditions, tectonics, and extinction events all influence their distribution. Modern tropical coral reefs are geologically young at their surfaces. Many grew during the period following the last glacial maximum. About 20,000 years ago, much of Earth's water was stored in continental ice sheets. Global sea level was more than 100 meters lower than today. As the ice sheets melted, sea level rose. Shallow tropical shelves flooded. Corals colonized suitable surfaces and began building upward. Many present reefs therefore rest on older limestone, volcanic rock, or submerged coastal landscapes. Their visible living layer may be thin compared with the total thickness of the underlying reef structure. Some reef deposits are hundreds or thousands of meters thick. These thick sequences accumulated over very long periods while the seafloor subsided or sea level changed. The formation of coral atolls helped shape early theories of reef development. Charles Darwin proposed a subsidence theory after observations made during the voyage of the Beagle. Darwin suggested that reefs begin as fringing reefs around volcanic islands. As an island slowly subsides, the coral continues growing upward. A channel develops between the reef and the land, producing a barrier reef. If the island eventually sinks below sea level, a ring-shaped atoll remains around a central lagoon. This general model was later supported by drilling and geological evidence. However, real reef development is often more complicated. Sea level changes can expose reefs, drown them, or create several generations of reef growth. Erosion and sedimentation also modify their shapes. Now let us move slowly from the individual coral animal to the structure of an entire reef. A fringing reef grows directly along a shoreline or close to it. It may have a narrow, shallow lagoon between the reef and the land. A barrier reef lies farther offshore. A broader and deeper lagoon separates it from the coast. An atoll is a roughly ring-shaped reef enclosing a lagoon. It may contain low islands made of reef sediment. Patch reefs are smaller, isolated reef structures. They occur within lagoons, on shelves, and between larger reef systems. Bank reefs and platform reefs rise from continental or oceanic shelves. They may not be directly associated with an island. Ribbon reefs are long, narrow structures. They are especially well developed along parts of the Great Barrier Reef. Reef classification can be based on shape, position, geology, hydrodynamics, or biological community. The same reef may fit several descriptive categories. A typical reef has several environmental zones. The exact arrangement depends on wave exposure, depth, tides, local geology, and region. The reef flat is a shallow, relatively horizontal area. During low tide, parts of it may be close to the water's surface or temporarily exposed. The reef crest is often the highest energy zone. Waves break over it. Corals and coralline algae living there must tolerate strong water movement. The forereef, or reef slope, faces the open sea. It descends toward deeper water. Light decreases with depth, and coral communities change accordingly. The back reef lies on the sheltered side. It may contain seagrass beds, patch reefs, sand, rubble, or mangrove habitats. Lagoons are protected areas behind barriers or within atolls. Their depth and water circulation vary widely. The shallowest reef crest may be dominated by organisms adapted to breaking waves. Encrusting coralline algae can bind the surface. Robust branching or low-profile corals may occupy slightly deeper areas. On exposed reefs, repeated wave force influences colony shape. Compact colonies create less drag than delicate forms. Flexible organisms bend rather than break. Wave energy also brings oxygen and food. It removes sediment and waste products. Moderate water flow can increase coral growth. Very strong flow can damage tissue or break skeletons. Very weak flow can allow sediments to settle and reduce gas exchange. Water movement around coral colonies is complex. Each branch, ridge, and polyp changes the local flow. Thin boundary layers form near surfaces. Inside the boundary layer, molecular diffusion becomes important. Oxygen, carbon dioxide, nutrients, and waste products must cross this layer. Faster flow usually makes the boundary layer thinner. This improves transport between coral tissue and seawater. At night, oxygen can fall inside dense coral branches because photosynthesis is stopped while respiration continues. Water movement helps prevent extreme oxygen depletion. Light is one of the main factors controlling coral distribution with depth. Water absorbs and scatters light. Red and orange wavelengths disappear relatively quickly. Blue light penetrates deeper into clear ocean water. Photosynthetic symbionts adjust to different light conditions. In low light, they may increase pigment concentrations or change the organization of photosynthetic membranes. Corals in shallow water must manage excess light. Strong ultraviolet radiation and high visible light can damage cells, especially during heat stress. Protective pigments help reduce this damage. Some corals produce fluorescent proteins and chromal proteins. These molecules absorb or transform light. Fluorescent proteins may emit green, cyan, red, or other wavelengths after absorbing higher energy light. Their functions vary and may include photoprotection, light modulation, antioxidant activity, and cellular signaling. Coral tissue can appear brown because of the photosynthetic pigments inside symbiotic algae. Other colors come from coral host pigments and skeletal reflection. A healthy coral is not always brightly colored. Some healthy species are pale brown, gray, cream, or muted green. Corals growing in low light may have darker tissue because they contain more photosynthetic pigment. Shallow corals may appear lighter or contain stronger protective pigments. Depth limits vary greatly. Most reef construction occurs in the upper few tens of meters, where light is abundant. However, photosynthetic corals can live much deeper in exceptionally clear water. Mesophotic coral ecosystems generally occur from about 30 meters to more than 100 meters deep. The term mesophotic means middle light. These reefs receive limited but usable sunlight. Mesophotic reefs may contain plate-like corals, sponges, algae, and specialized fish communities. Their species composition often differs from shallow reefs. Deep mesophotic reefs are not necessarily safe refuges for shallow water species. Some species occur across broad depth ranges, but many do not. Conditions at depth include lower light, cooler temperatures, different currents, and altered food supply. The vol exchange between shallow and deep populations may be limited. Below the photic zone, non-photosynthetic corals can still live. Cold water corals occur in deep oceans around the world. Cold water corals do not depend on algal symbionts. They capture food from currents. Some create large three-dimensional reef structures in cold, dark water. Species such as Laphelia pertusa, now also called Desmophyllum pertusum in revised classifications, can form extensive deep-sea reefs. These reefs may occur hundreds or thousands of meters below the surface. They grow slowly and can be very old. The science of tropical coral reefs and cold water coral ecosystems overlaps, but their energy sources and environmental controls differ substantially. Tropical reef corals usually live between about 23 degrees north and 23 degrees south, although reefs extend beyond these latitudes in some regions. Temperature is an important limit. Reef-building corals generally grow best in warm water. Many reefs occur where average temperatures remain between about 23 and 29 degrees Celsius. Local populations can exist outside this range. Some high-latitude reefs experience winter temperatures below 18 degrees Celsius. Certain equatorial regions occasionally exceed 30 degrees Celsius. The important factor is not only absolute temperature. Corals are adapted to their local seasonal range. A temperature that is tolerable on one reef may cause stress on another. Sudden change can be more damaging than gradual change. Most tropical reefs occur in saline marine water. Very low salinity can damage corals. Heavy rainfall, river discharge, and flooding may create low salinity surface layers. Shallow corals can experience osmotic stress if these layers persist. Corals differ in their tolerance. Species from lagoons and nearshore habitats often withstand greater variation than species from stable offshore environments. Sediment also influences reef distribution. Suspended sediment reduces light. Settled sediment can cover living tissue. Corals use cilia, mucus, tentacle movement, and tissue expansion to remove particles. These processes require energy. Some species can remove sediment efficiently. Others are quickly damaged by even a thin layer. Fine sediment is often more harmful than coarse sediment because it remains suspended longer and penetrates small surface spaces. Sediment may also carry nutrients, metals, pesticides, petroleum compounds, or microorganisms. Nearshore reefs naturally experience some sedimentation. Problems become more severe when land clearing, dredging, construction, mining, or agriculture greatly increases the sediment supply. Reefs also require suitable substrate. Coral larvae generally need a stable, hard surface. Loose sand ships too easily for most coral settlement. Rubble can provide hard surfaces, but unstable rubble may roll during waves and crush young colonies. Crustose coralline algae help stabilize surfaces. Microbial films develop on submerged rock and influence larval settlement. Bioerosion creates another important part of reef structure. Bioerosion is the biological breakdown of calcium carbonate. Parrotfish scrape or excavate reef surfaces while feeding on algae. Sea urchins graze with hard teeth. Sponges bore into skeletons. Worms, bivalves, and microorganisms also remove carbonate. Some bioeroders produce recognizable tunnels and holes. Others weaken the skeleton at microscopic scales. Bioerosion is not purely destructive. It creates sand, opens habitat, recycles carbonate, and prevents some surfaces from being completely covered. A functioning reef exists in balance between construction and erosion. Coral growth and mineral deposition add material. Grazing, boring, dissolution, and storms remove it. When construction exceeds loss, the reef framework grows. When loss exceeds construction, the reef begins to flatten or shrink. The reef carbonate budget is a calculation of these gains and losses. Researchers estimate coral calcification, coralline algal growth, sediment production, bioerosion, and physical export. Positive carbonate budgets indicate net accumulation. Negative budgets indicate net loss. The carbonate budget can become negative before the reef appears completely dead. A reef may still contain living corals while losing structural complexity. Structural complexity is important because cracks, branches, holes, and overhangs provide shelter. Many fish and invertebrates depend on these spaces. As a reef erodes, habitat becomes flatter. Large predators, small sheltering fish, juvenile organisms, and cryptic species may all be affected. Storms can remove large amounts of carbonate in a short time. Waves break bridges, overturn colonies, and transport rubble. The effect of a storm depends on intensity, duration, direction, reef orientation, and coral form. The same storm may heavily damage one area and leave another largely unchanged. Storms also create new settlement surfaces. Broken fragments may survive. Rubble movement can expose clean substrate. In healthy systems, coral recovery may follow disturbance. Recovery can take years or decades. Repeated disturbances reduce the time available for recovery. If severe events occur too frequently, the community may shift to a different state. Reef growth occurs vertically and laterally. Vertical growth helps the reef remain within the lighted zone as sea level rises or the foundation subsides. If sea level rises slowly, many reefs can keep pace. If it rises faster than reef accretion, the reef may deepen. A reef that cannot maintain sufficient light for its corals may drown. Drowned reefs can remain as submerged carbonate platforms. During sea level fall, the reef surface may be exposed to air. Rainwater dissolves limestone and forms caves, pits, and channels. These exposed surfaces can later be flooded again. New coral growth develops over the eroded foundation. Reef limestone is porous. Water moves through cracks and cavities. Chemical exchange occurs between seawater, groundwater, sediments, and pore spaces. At night, respiration releases carbon dioxide. Carbon dioxide lowers pH and can promote dissolution within reef sediments. During the day, photosynthesis removes carbon dioxide and raises pH. Calcification removes carbonate and can lower total alkalinity. The chemistry of reef water therefore changes over a daily cycle. Dissolved oxygen usually rises during daylight because of photosynthesis. It falls at night because organisms continue to respire. In calm pools, nighttime oxygen can become very low. Daytime oxygen can become supersaturated. The pH may also vary substantially. Some reef flats experience daily pH changes much larger than those of the open ocean. These natural fluctuations do not mean reefs are protected from long-term ocean acidification. Short-term variability and long-term baseline change are different processes. Ocean acidification begins when seawater absorbs carbon dioxide from the atmosphere. Carbon dioxide reacts with water to form carbonic acid. Carbonic acid dissociates into bicarbonate and hydrogen ions. The increase in hydrogen ions lowers pH. It also converts some carbonate ions into bicarbonate. Coral calcification depends on the availability of carbonate ions. Lower carbonate concentration reduces the saturation state of aragonite. Aragonite saturation state describes how favorable seawater chemistry is for aragonite formation and persistence. When the saturation state is above 1, seawater is supersaturated with respect to aragonite. This means aragonite formation is chemically possible and existing aragonite tends not to dissolve spontaneously in open water. Most tropical surface seawater currently remains supersaturated. However, declining saturation state can still make biological calcification more difficult. Corals can regulate the chemistry of their calcifying fluid, so their response is not determined only by surrounding seawater. Some species maintain calcification under lower pH better than others. They may spend more energy controlling internal chemistry. Ocean acidification can also weaken reef cementation, increase dissolution, alter larval development, and affect algae, mollusks, echinoderms, and plankton. The combined effect on reef structure may be greater than the effect on living coral calcification alone. Reef sediments are made from many sources. Coral fragments contribute. Coralline algae contribute. Mollusk shells, foraminifera, echinoderm skeletons, and fish-produced carbonate also contribute. Parrotfish create sediment when they grind carbonate while feeding. Much of the material passes through the digestive system and is released as fine sand. The amount produced by an individual fish depends on species, size, diet, feeding mode, and habitat. Some tropical beach sand comes from parrotfish, but reef sand has many origins. It should not be attributed to one organism alone. Green algae in the genus Halimeda can also produce large amounts of carbonate. Their segmented tissues contain calcium carbonate crystals. When Halimeda segments die, the organic material breaks down and carbonate fragments enter the sediment. Foraminifera are single-celled organisms with shells called tests. Some reef-associated foraminifera host photosynthetic symbionts and grow relatively large. Their tests accumulate in sediments and may form a substantial fraction of tropical beach material. Sediments are transported by waves and currents. They fill cavities, build islands, form sand cays, and move between reef zones. Reef islands are dynamic structures. Their shorelines can shift as storms and currents rearrange sediment. Some islands migrate across reef platforms over decades or centuries. Others remain relatively stable because vegetation and reef structure trap material. The existence of a reef island depends on the balance between sediment production, transport, erosion, and sea level. Coral reefs influence wave energy. The reef crest causes waves to break. Friction across the shallow reef flat reduces their height. This process protects many coastlines from erosion and flooding. The degree of protection depends on reef width, depth, roughness, continuity, and water level. A healthy reef with high structural complexity usually dissipates more wave energy than a flattened reef. During extreme storms, reefs cannot prevent all flooding. They can still reduce incoming wave force. The protective service of reefs has economic value because many coastal settlements and infrastructures lie behind them. Now let us drift from reef structure into the broader ecological community that occupies it. Coral reefs support organisms from nearly every major marine animal group. They contain fishes, crustaceans, mollusks, echinoderms, sponges, worms, tunicates, bryozoans, cnidarians, and many smaller forms. Algae are also central. They include microscopic phytoplankton, turf algae, crustose coralline algae, fleshy macroalgae, and endolithic algae living inside rocks. Bacteria and archaea occupy water, mucus, sediments, animal tissues, and mineral surfaces. Viruses infect organisms throughout the community. The total number of reef species is uncertain. Many small organisms have not been formally described. Cryptic species live inside skeletons, under rubble, within sponges, or between sediment grains. They are easy to overlook. A single piece of coral rubble may contain small crabs, shrimp, worms, mollusks, brittle stars, sponges, and microbial films. Biodiversity varies among regions. The highest coral diversity occurs in the Indo-Pacific, especially within the Coral Triangle. The Coral Triangle includes marine areas around Indonesia, the Philippines, Malaysia, Papua New Guinea, Timor-Leste, and the Solomon Islands. This region contains several hundred reef-building coral species and extremely high fish diversity. The Caribbean contains fewer coral species than the Indo-Pacific. Its evolutionary and geological history is different. The closure of the Central American Seaway separated Atlantic and Pacific populations several million years ago. Later, climate changes and extinctions removed many Caribbean coral lineages. The modern Caribbean reef community therefore has lower taxonomic diversity, but remains ecologically important. Coral reefs are often described as the rainforests of the sea. The comparison refers to biodiversity, structural complexity, and dense ecological interactions. However, reefs function differently from terrestrial forests. Most reef biomass is animal, algal, microbial, or mineral-associated rather than wood-based. Primary production on reefs comes from several sources. Coral symbionts contribute. Turf algae contribute. Macroalgae, seagrasses, phytoplankton, and microorganisms also contribute. Gross primary production can be high. Net production may be lower because reef communities also have high respiration. A large fraction of organic matter is rapidly recycled. Nutrients pass repeatedly through microbes, algae, corals, sponges, and consumers. This recycling helps explain the apparent paradox of productive reefs in nutrient-poor water. The open tropical ocean around many reefs contains little nitrogen and phosphorus, yet the reef itself concentrates organisms and maintains rapid nutrient turnover. Water moving over the reef delivers plankton, dissolved compounds, and particles. The reef removes some of this material. At the same time, reef organisms release mucus, larvae, waste, gametes, and dissolved organic matter. The boundary between reef and open ocean is therefore active. Material moves in both directions. Sponges play a major role in this exchange. Many sponges filter large volumes of water. They remove bacteria, dissolved organic matter, and small particles. Some convert dissolved organic matter into cellular material that is shed and consumed by other organisms. This process is sometimes called the sponge loop. It helps retain energy within the reef food web. Sponges also host dense microbial communities. These microbes perform photosynthesis, nitrogen fixation, nitrification, denitrification, and other chemical processes. Some sponges bore into carbonate. Others add three-dimensional habitat. Microbial processes occur rapidly in reef sediments. Oxygen penetrates only a short distance below the surface in many locations. Below the oxygenated layer, microbes use alternative chemical reactions. They may reduce nitrate, sulfate, iron, or other compounds. Sulfate reduction produces hydrogen sulfide. Other microbes can oxidize this sulfide when oxygen becomes available. Nitrogen gas can be produced through denitrification and dynamics. Nitrogen can also enter the system through biological nitrogen fixation. Nitrogen-fixing microorganisms convert atmospheric nitrogen gas into biologically usable compounds. Some cyanobacteria fix nitrogen, Symbiotic bacteria in sponges, corals, and algae may also contribute. The net nitrogen balance of a reef depends on fixation, denitrification, export, and external inputs. Phosphorus can bind to particles or minerals. It can be released during decomposition and reused. Iron is often scarce in clear ocean water. Dust, land runoff, upwelling, and sediments can supply it. Trace elements may limit or stimulate different organisms. Excess concentrations can become toxic. Reef food webs are highly connected. Organisms often change diet with age, season, or opportunity. Many reef fishes are omnivores. A fish classified as herbivorous may also consume detritus, microbes, or small animals. Grazing fishes influence competition between corals and algae. Parrotfish and surgeonfish remove algal tissue from reef surfaces. Some species scrape the surface. Others crop soft algal filaments. Large excavating parrotfish remove pieces of carbonate. Rabbitfish, damselfish, chubs, and other groups also consume algae. Herbivores do not all perform the same ecological role. One species may remove short turf algae, another may consume large fleshy macroalgae. A reef with many herbivore species often has broader grazing capacity than a reef dominated by one type. Sea urchins can also be major grazers. In the Caribbean, the long-spined sea urchin Diadema antillarum was once extremely abundant. During the early 1980s, a disease caused mass mortality across much of the Caribbean. The decline reduced grazing pressure. Algae increased on many reefs, especially where herbivorous fishes were also depleted. The event contributed to major ecological changes. Although it was not the only cause, predatory fishes include groupers, snappers, jacks, barracudas, moray eels, and reef sharks. Predators influence prey abundance and behavior. Their effects can spread through the food web. The strength of these effects varies. Reef ecosystems do not always follow a simple chain from predator to herbivore to algae. Fishing can remove large predators, herbivores, or both. Selective fishing changes size, structure, and species composition. Large fishes often have high reproductive output. Removing them can reduce population resilience. Some fishes change sex during their lives. Many wrasses and parrotfishes are sequential hermaphrodites. In protogynous species, individuals begin life as females, and some later become males. In protandrous species, individuals begin as males and later become females. Social structure can influence sex change. Removing the largest individuals may therefore affect reproduction in ways that are not obvious from total fish numbers alone. Reef fishes use different habitats during their life cycles. Juveniles may live in mangroves or seagrass beds. Adults may move to coral reefs. Mangrove roots provide shelter from large predators. Seagrass meadows offer food and nursery habitat. The ecological connection among reefs, mangroves, and seagrasses can increase fish abundance and diversity. Not all reef species require these neighboring habitats. The strength of connection differs among regions and species. Fish movement also transports nutrients. A fish may feed in one habitat and release waste in another. Seabirds can deliver nutrients from the open ocean to reef islands. Their guano contains nitrogen and phosphorus. These nutrients can enter surrounding coastal water. They may increase productivity, although excessive enrichment can also create imbalances. Reef organisms compete for space because hard surface is limited. Corals can overgrow one another. They can shade neighbors. They may extend digestive filaments or specialized tentacles. Sweeper tentacles are elongated tentacles used in competition. They contain many nematocysts and can damage nearby tissue. Some corals extrude mesenterial filaments. These digestive tissues contact and digest neighboring organisms. Competition occurs slowly. A boundary between colonies may remain nearly unchanged for months. Sponges can overgrow corals or release chemicals. Algae can shade tissue, trap sediment, or cause abrasion. Corals can also resist algae through mucus, tissue movement, chemical compounds, and help from grazing animals. The outcome depends on water quality, herbivory, growth rate, disturbance, and species identity. Corals contain many chemical compounds. Some deter predators. Some prevent microbial growth. Some mediate competition. Soft corals, which generally do not build massive stony skeletons, are especially known for producing defensive chemicals. Soft corals belong mainly to the subclass Octocoralia. Their polyps usually have 8 tentacles with small side branches. Stony corals belong to Hexacoralia. Their body structures are often arranged in multiples of 6. Octocorals may contain small skeletal elements called sclerages. These provide support and defense. Gorgonians, sea fans, sea pimps, and sea pens are octocorals. Many are common on tropical reefs. Sea fans orient their branches across prevailing currents. This position improves capture of plankton. Zoantids and sea anemones are other anthozoans found on reefs. Hydrocorals, such as fire corals, resemble stony corals but belong to the class Hydrozoa. Fire corals form hard skeletons and contain powerful stinging cells. They are not true scleractinian corals. Mallepora species are important reef builders in some regions. Their branching or encrusting colonies can occupy wave-exposed areas. Other cnidarians include corallimorpharians, which resemble anemones and corals but lack large external skeletons. Corallimorpharians can sometimes spread rapidly over disturbed reef surfaces. Jellyfish are less permanently associated with reefs, but some species use lagoons and coastal habitats during parts of their life cycles. Mollusks are extremely diverse on reefs. Gastropods include grazers, predators, scavengers, and parasites. Bivalves filter particles from water. Giant clams host photosynthetic symbionts in their tissues. Giant clams belong to the family Cardiidae, subfamily Tridacninae. They include several species in the genera Tridacna and Hippopus. Their mantles contain symbiotic dinoflagellates. Light supports photosynthesis. The clam also filter feeds. The shell continues growing throughout life. Some species become more than 1 meter long. Giant clams contribute carbonate and provide habitat on their shell surfaces. Cone snails are predatory gastropods. They inject venom using a modified radular tooth. Different cone snail species hunt worms, mollusks, or fish. Their venoms contain many biologically active peptides. Most cone snails do not pose a serious danger to humans, but a few fish-hunting species can cause severe envenomation. Nudibranchs are shell-less marine gastropods. Many display bright colors. Some feed on sponges, hydroids, anemones, or soft corals. They may store defensive chemicals or nematocysts from their prey. Bivalves boring into coral skeletons create cavities. Some remain completely enclosed except for siphon openings. Crustaceans occupy nearly every reef microhabitat. They include crabs, shrimp, lobsters, amphipods, copepods, barnacles, and isopods. Many coral colonies host small crabs and shrimp. Some defend the coral from predators or remove sediment. Certain guard crabs live among branching corals. They may attack coral-eating sea stars or snails. In return, the coral provides shelter and mucus. Cleaner shrimp remove parasites and dead tissue from fish. They often occupy fixed cleaning stations. Fish approach these stations and adopt characteristic postures. The interaction provides food for the cleaner and hygiene for the client. Cleaning is not always perfectly cooperative. Cleaners may sometimes consume mucus or tissue rather than parasites. Client fish can respond by leaving, chasing the cleaner, or using a different station. Cleaner wrasses perform similar services. Some species mimic cleaner wrasses to approach other fish. The blue-striped fangblenny resembles a cleaner wrasse and may bite scales or tissue from deceived fish. This is an example of aggressive mimicry. Other reef organisms use defensive mimicry. Harmless species may resemble toxic or venomous ones. Camouflage is also widespread. Stonefish, scorpionfish, octopuses, flatfish, and crustaceans can match reef backgrounds. Cephalopods change color using pigment cells called chromatophores. Reflective cells called iridophores and leucophores modify light. Octopuses can also change skin texture. Mussels raise small structures called papilla. These changes allow rapid camouflage and communication. Reef octopuses are intelligent predators. They eat crabs, mollusks, and other animals. Some carry shells or pieces of material for shelter. Others occupy holes in coral rubble. Echinoderms include sea stars, riddle stars, sea urchins, sea cucumbers, and crinoids. Sea cucumbers and crinoids. Sea cucumbers consume sediment or suspended particles. They process organic material and release cleaned sediment. Their feeding changes sediment structure and chemistry. Some species increase oxygen penetration and nutrient exchange. Sea cucumbers can eject sticky internal structures or defensive chemicals when threatened. Sea stars have a water vascular system. Tube feet operate through hydraulic pressure. Some sea stars prey on bivalves. Others consume detritus, sponges, or corals. The crown-of-thorns sea star feeds on living coral tissue. It occurs naturally in the Indo-Pacific. At low density, crown-of-thorns sea stars are part of normal reef ecology. At high density, outbreaks can remove large areas of coral. The causes of outbreaks are still studied. Possible factors include nutrient-enhanced larval survival, removal of predators, oceanographic conditions, and natural population cycles. Adult crown-of-thorns sea stars can consume several square meters of coral tissue per year, depending on size and conditions. They often prefer fast-growing branching or table corals. When those become scarce, they consume other species. Some corals resist attack through aggressive crabs, thick tissue, or strong nematocysts. Resistance is not complete. Triton snails, pufferfish, triggerfish, and other predators may consume crown-of-thorns sea stars or juveniles. Control programs sometimes remove or inject individual sea stars in heavily affected areas. Local control can protect small, high-value sites. It is difficult to manage outbreaks across entire reef systems. Brittle stars hide in crevices and extend arms to capture particles. Some form dense groups on sponges or corals. Crinoids, also called feather stars, use branching arms to filter food from currents. They often emerge at night and hide during the day. Reef worms are diverse. Polychaete worms may crawl, burrow, build tubes, filter feed, graze, or prey on other animals. Christmas tree worms live in tubes embedded within coral skeletons. Their spiral crowns are used for feeding and gas exchange. When disturbed, they rapidly withdraw into the tube. A small operculum closes the opening. The worm does not usually kill the entire coral colony. The relationship can persist for years. Fireworms have hollow bristles containing irritating substances. They may feed on corals, anemones, carrion, or small animals. Flatworms occur on reef surfaces and sediments. Some are brightly colored and resemble nudibranchs. Sponges are among the oldest animal lineages. They lack true organs and nervous systems. Water enters through small pores, passes through internal canals, and exits through larger openings. Specialized cells, called colwinocytes, create water currents and capture food. Sponges can filter particles smaller than those captured by many other animals. Their skeletons may contain silica spicules, calcium carbonate spicules, or flexible spongin fibers. Some species contain all three structural components in different combinations. Sponges reproduce sexually and asexually. Many release swimming larvae. They also regenerate after fragmentation. A small surviving piece can sometimes produce a new individual. Sponges compete chemically and physically. Some release compounds that discourage settlement or growth of neighbors. Many sponge compounds have been investigated for pharmaceutical uses. A few have inspired antiviral, anti-cancer, or immunosuppressive drugs. The compound itself is not always produced by the sponge. Symbiotic microbes may be responsible. Bryozoans are colonial animals with tiny feeding units called zooids. Each zooid extends a crown of tentacles called a lophore. It filters small particles from water, Bryozoan colonies can be encrusting, branching, or lace-like. Many produce calcium carbonate skeletons. Tunicates are chordates. Their larvae have a notochord, although adults often look unlike other chordates. Sea squirts filter water through a perforated internal basket. Some are solitary. Others form colonies across reef surfaces. Colonial tunicates can spread rapidly. Their soft tissues may contain defensive chemicals. Fish diversity is one of the most visible features of coral reefs. Reef fishes belong to many evolutionary lineages. The term reef fish describes habitat use rather than a single taxonomic group. Rosses are among the largest reef fish families. They show wide variation in color, size, diet, and behavior. Some wrasses remove parasites. Some crush mollusks. Some feed on plankton or small invertebrates. Parrotfishes are closely related to wrasses. Their front teeth are fused into a beak-like structure. They scrape algae and microorganisms from hard surfaces. Powerful pharyngeal teeth grind ingested material. Surgeonfishes have sharp spines near the base of the tail. These spines may be fixed or movable. Most surgeonfishes graze algae or consume plankton. Their digestive systems contain microbes that help process food. Damselfishes are often small and territorial. Some cultivate patches of turf algae. They remove unwanted algae and defend the territory from grazers. These farming damselfishes can alter local coral recruitment. Their territories may contain dense algal mats. Other damselfishes feed on plankton above branching corals. They retreat into branches when predators approach. Anemonefishes live among the tentacles of sea anemones. A mucus coating helps prevent the anemone from discharging nematocysts against the fish. The fish gains protection. The anemone may receive nutrients, water movement, defense, and food scraps. Anemone fish groups have a social hierarchy. The largest individual is female. The next largest is the breeding male. If the female disappears, the breeding male changes sex and becomes female. A smaller fish becomes the breeding male. Butterfly fishes often have narrow bodies and small mouths. Many feed on coral polyps, small invertebrates, or plankton. Some species are obligate corallivores and depend heavily on live coral. Their abundance may decline after coral loss. Other butterfly fishes have broader diets and are more flexible. Angelfishes feed on sponges, algae, tunicates, and other organisms. Their laterally compressed bodies allow movement through complex reef structure. Groupers are ambush predators with large mouths. They create suction to capture prey. Many groupers form spawning aggregations at predictable sites and seasons. These aggregations can contain hundreds or thousands of fish. They are vulnerable to intensive fishing. Protecting spawning sites can improve reproduction, but enforcement and local cooperation are necessary. Reef sharks include gray reef sharks, blacktip reef sharks, whitetip reef sharks, Caribbean reef sharks, and others. Different species use reefs in different ways. Some remain near a small area. Others travel between islands or offshore habitats. Sharks generally reproduce slowly. Many mature late and produce few young. This makes their populations vulnerable to high fishing mortality. The ecological role of sharks differs among reefs. They may regulate prey behavior, remove injured animals, and connect habitats through movement. Evidence for strong trophic cascades varies among systems. It should not be assumed that every shark decline produces the same result. Moray eels occupy crevices. Their long bodies allow them to hunt inside complex structures. They use a second set of jaws in the throat, called pharyngeal jaws. These jaws move forward to grasp prey and pull it into the esophagus. Some groupers and moray eels hunt cooperatively. The grouper chases prey toward crevices while the moray enters the crevice. Cooperative hunting is not universal, but it has been documented in several locations. Reef fishes also produce sounds. They grind teeth, vibrate, swim bladders, snap tendons, or move bones. Sound is used in courtship, aggression, territorial defense, and group coordination. A coral reef has a characteristic soundscape. Snapping shrimp create continuous crackling. Fish produce lower frequency calls. Waves and water movement add background noise. Larval fishes and invertebrates may use reef sound to locate habitat. Healthy and degraded reefs can have different acoustic patterns. Artificial playback experiments suggest that reef sound can influence settlement, but responses vary among species. Reef sound travels farther at night under some conditions. Temperature, depth, bottom shape, and biological activity affect transmission. If you are still listening, there is nothing you need to remember. The terms can simply pass by at their own steady pace. Many reef organisms are active mainly at night. Day-active fishes enter shelters. Nocturnal predators emerge. Squirrelfish and soldierfish leave crevices to feed. Cardinalfish move into open water. Plankton rises from deeper water. Coral polyps extend. Crustaceans emerge from holes. The change between day and night restructures the food web every 24 hours. At dawn, nocturnal species return to shelter. Day-active grazers and plankton feeders resume activity. Some predators exploit these transition periods. Reef organisms also follow lunar cycles. Spawning, migration, feeding, and settlement may be synchronized with moonlight or tides. Tidal range differs among reefs. Some experience changes of several meters. Others have much smaller tides. Tides alter depth, current speed, temperature, salinity, and access to habitat. Intertidal corals may be exposed to air during low tide. They must tolerate heating, ultraviolet radiation, desiccation, and fresh rainwater. Some retract their polyps and produce mucus. Others occupy pools that remain submerged. Exposure tolerance differs among species and populations. Reef flats can reach very high temperatures during calm, low tides. They can also cool rapidly at night. Organisms living there often experience greater daily variation than those on deeper slopes. This environmental variability may increase tolerance to some stresses. It can also create repeated energetic costs. Coral reef ecology includes many mutualisms. Gobies live with burrowing shrimp. The shrimp excavates and maintains a shelter. The goby watches for predators. It maintains contact with the shrimp using its tail. When the goby signals danger, both animals retreat into the burrow. Cleaner interactions are another mutualism. Corals and guard crabs form another. Some fishes remove sediment from corals while feeding. Some sea cucumbers shelter small fish or crustaceans inside their bodies. Mutualisms are not always equal at every moment. The costs and benefits change with conditions. Commensal relationships benefit one partner without a clear effect on the other. Parasitic relationships benefit one organism while harming another. Reef parasites include worms, crustaceans, protozoans, fungi, bacteria, and viruses. Many fish carry ectoparasitic copepods or isopods. Some parasites live inside organs or muscles. Parasites are part of natural biodiversity. They influence food webs and host evolution. Disease becomes a population-level problem when infection spreads widely or mortality rises above normal background levels. Coral diseases are identified through tissue loss patterns, discoloration, lesions, growth abnormalities, and laboratory tests. Names such as black band disease, white band disease, white band disease, white plague, yellow band disease, and stony coral tissue loss disease describe different syndromes. A visible disease sign does not always reveal a single pathogen. Similar lesions can have different causes. Black band disease involves a microbial mat containing cyanobacteria and other microorganisms. The band moves across a colony, leaving exposed skeleton behind. White band disease caused major declines of Caribbean acropora corals during the late 20th century. Its exact causal agents remain uncertain. Stony coral tissue loss disease was first documented near Florida in the 2010s. It spread across much of the Caribbean. It affects many stony coral species and can cause rapid tissue loss. Researchers have investigated bacterial involvement, viral particles, environmental factors, and host responses. Antibiotic pastes can slow lesions in treated colonies. This suggests bacteria play an important role, although the full disease process remains under study. Disease risk often increases when corals are stressed by high temperature, pollution, sediment, or physical damage. Warmer water can accelerate pathogen growth and weaken host defenses. Not all coral mortality is caused by infectious disease. Predation, bleaching, sediment, low salinity, chemical exposure, and competition can produce similar, visible damage. Coral immunity relies on cellular and chemical defenses. Corals lack the antibody-based adaptive immune system found in vertebrates. They possess complex innate immune pathways. Immune cells recognize molecular patterns associated with damage or microbes. Responses include phagocytosis, melanin production, antimicrobial compounds, programmed cell death, and wound repair. Melanin can form dark deposits around damaged tissue. It helps isolate pathogens and strengthen repair areas. Corals also maintain mucus barriers. The mucus contains microbes, enzymes, and chemical compounds. A stable microbial community may prevent pathogens from establishing. This idea is sometimes compared with colonization resistance in other animals. Coral wound healing occurs through tissue migration and skeletal growth. Small injuries may close completely. Large injuries are more likely to become infected or overgrown by algae. Energy availability affects healing. A well-fed coral may repair damage more effectively. Reproduction can decline after stress because energy is redirected towards survival and repair. Some colonies survive bleaching but produce fewer gametes in the following season. Stress can therefore influence population recovery before adult mortality becomes visible. Bleaching is one of the best-known coral stress responses. A bleached coral appears pale or white because it has lost symbiotic algae, algal pigments, or both. The white calcium carbonate skeleton becomes visible through translucent tissue. Bleaching is not the same as death. The coral animal may remain alive. If stressful conditions end soon enough, symbiont populations can recover. If stress is severe or prolonged, the coral may starve, become diseased, or die. Heat stress is the most widespread cause of mass coral bleaching. When water remains unusually warm, photosynthesis becomes disrupted. Excess energy produces reactive oxygen species in algal and coral cells. Reactive oxygen species can damage proteins, lipids, membranes, and deoxyribonucleic acid. The symbiosis becomes unstable. Algal cells may be expelled, digested, or lost when host cells detach. Several cellular pathways may operate at the same time. Bleaching can also be caused by cold, excessive light, darkness, pollution, low salinity, or disease. However, repeated large-scale bleaching events are mainly associated with marine heat waves. Scientists often measure accumulated heat stress using degree heating weeks. 1 degree heating week represents 1 degree Celsius above a defined breaching threshold sustained for 1 week. It can also represent 2 degrees above the threshold for half a week, or another equivalent combination. The threshold is commonly based on the highest expected monthly mean temperature for a location. Moderate accumulated heat stress can cause bleaching. Higher values increase the likelihood of widespread bleaching and mortality. These relationships are statistical rather than absolute. Local currents, cloud cover, wind, species composition, and prior exposure alter outcomes. Strong sunlight can intensify heat stress. Calm weather allows surface water to warm and reduces mixing. Clouds and storms may lower light and temperature, sometimes reducing bleaching severity. Turbid water can also reduce light, although sediment brings other stresses. Coral species differ in bleaching sensitivity. Branching Acropora and Pochilopora species are often sensitive. Some massive porite species may resist bleaching longer. Although they are not immune, within one species, colonies can differ because of genetics, symbionts, depth, microhabitat, nutrition, and stress history. Bleaching patterns may vary across a single colony. Shaded surfaces can retain more symbionts than sun-exposed surfaces. Fluorescent pigments can become more visible during bleaching. Some corals develop bright pink, purple, or yellow coloration. This phenomenon is sometimes called colorful bleaching. The colors may result from increased host pigment production in areas where symbionts are reduced, but tissue remains alive. Colorful bleaching does not guarantee recovery. It indicates a particular tissue response. After a bleaching event, surviving corals may regain symbionts from remaining cells or from the environment. The symbiont community may change. A heat-tolerant type can become more abundant. This process is sometimes called symbiont shuffling, when existing types change in proportion. Symbiont switching refers more strictly to acquiring new types from outside the colony. Changes in symbionts can improve future heat tolerance. They may also reduce growth or reproduction. Bleaching affects the entire reef community. Coral-dependent fishes lose shelter and food. Dead coral skeletons may remain standing for months or years. At first, structural habitat can persist. Algae and microbes colonize the exposed skeleton. Bioeroders weaken it. Storms then break the skeleton into rubble. Structural complexity declines. This delayed erosion means ecological effects continue long after the original heatwave. Mass bleaching events were once considered unusual. They have become more frequent as ocean temperatures rise. The interval between severe events has shortened in many regions. When bleaching returns before corals mature and reproduce, recovery becomes difficult. Thermal stress also interacts with acidification. Lower pH can increase energetic demands. Nutrient pollution can increase bleaching sensitivity in some settings. Sediment reduces light but may also smother tissue and alter microbes. Disease frequently rises after heat stress. Physical disturbances can remove recovering colonies. The effects are cumulative. Reef resilience is the capacity to resist disturbance, recover afterward, or reorganize while retaining important functions. Resistance means the reef changes relatively little during stress. Recovery means coral cover, structure, and ecological processes return afterward. A reef can be resistant but slow to recover. It can also be sensitive yet recover rapidly if recruitment and growth are high. Resilience depends on coral diversity, herbivory, water quality, connectivity, substrate stability, disease, and disturbance frequency. No single metric fully describes it. Coral cover is commonly measured, but cover alone does not show colony size, species identity, growth, reproduction, or structural complexity. A reef with 20% cover of large framework building corals differs from a reef with 20% cover of small encrusting species. Fish biomass is useful, but it may hide the loss of particular functional groups. Recruitment counts young corals, but many recruits die before adulthood. Carbonate budgets provide information about long-term structure, disease prevalence, heat exposure, algal cover, water quality, and genetic diversity add further detail. Scientists monitor reefs using transects, quadrats, photographs, video, acoustic surveys, satellites, autonomous vehicles, and environmental sensors. A transect is a line placed across habitat. Organisms or conditions are recorded along it. A quadrat is a defined area used to measure cover, density, or abundance. Photoquadrats allow images to be analyzed later. Repeated images document change through time. Structure from motion photogrammetry combines overlapping photographs into three-dimensional models. These models measure surface area, colony volume, rugosity, and structural change. Diver surveys provide detailed observations but are limited by depth, time, and weather. Remote underwater vehicles can cover deeper or larger areas. Satellite sensors measure sea surface temperature, water color, turbidity, and broad habitat patterns. They cannot identify every coral species, but they provide frequent coverage over large regions. Airborne LiDAR uses laser pulses to measure shallow water depth and reef structure. Drones can map small, shallow areas at very high resolution. Acoustic methods use sound to estimate depth, bottom hardness, fish abundance, or habitat complexity. Environmental deoxyribonucleic acid, usually called environmental DNA, can detect genetic material released into water. A water sample may reveal the presence of fish, corals, microbes, or invasive species. Environmental DNA does not always show abundance accurately. Currents move genetic material away from its source. Genetic methods also identify coral species, symbionts, pathogens, and population connections. Traditional coral identification relies on skeletal form and colony appearance. This can be difficult because form changes with environment and closely related species may look similar. Molecular studies have revised coral classification. Some colonies once treated as one species have been divided. Other named species have been combined or moved into different genera. Taxonomy continues to change as more genomes and museum specimens are examined. Correct identification matters because species differ in growth, reproduction, disease sensitivity, and stress tolerance. Reef monitoring must account for observer error. Two divers may classify the same colony differently. Standard protocols, training, image archives, and automated analysis reduce inconsistency. Machine learning systems can identify benthic organisms in photographs. Their accuracy depends on image quality, training data, taxonomic resolution, and habitat diversity. Automated systems can process large data sets, but expert review remains important. Long-term monitoring is especially valuable. A single survey cannot distinguish temporary variation from long-term change. Historical photographs, ship records, fossils, sediment cores, and fisheries records extend the timeline. Older reef conditions are sometimes reconstructed from coral skeletons and reef cores. A drill core removes a cylinder of reef material. The layers show periods of growth, erosion, sedimentation, and exposure. Radiocarbon dating can estimate the age of material up to tens of thousands of years old. Uranium-thorium dating is used for many coral samples and can reach farther back with high precision under suitable conditions. These records show that reefs have responded to past climate changes. They also show that the present rate of atmospheric carbon dioxide increase is unusually rapid compared with many natural changes. Now let us move into the chemistry and physics that regulate reef life in more detail. Seawater contains dissolved salts. The most abundant ions include chloride, sodium, sulfate, magnesium, calcium, and potassium. Average open ocean salinity is about 35 grams of dissolved salts per kilogram of seawater. Salinity varies with evaporation, rainfall, river input, ice formation, and circulation. Warm surface water is usually less dense than cool water. Fresher water is less dense than saltier water. Density differences help drive ocean circulation. Tropical oceans often develop a warm surface layer separated from cooler deep water by a thermocline. A thermocline is a depth interval where temperature changes rapidly. Internal waves move along density boundaries. They can bring cool, nutrient-rich water toward reefs. Short pulses of cool water may reduce heat stress. They may also expose corals to low oxygen or lower pH. Upwelling brings deeper water toward the surface. This water is usually cooler and richer in nutrients and dissolved in organic carbon. Reefs near upwelling zones may experience high productivity and strong chemical variability. They may also be exposed to naturally lower pH. Some corals in variable environments show tolerance to fluctuating conditions. This does not mean they can tolerate unlimited change. Local adaptation can involve trade-offs. A population adapted to variable temperature may grow more slowly. Ocean currents transport heat, larvae, plankton, pollutants, and sediments. Large current systems influence the geographic range of reef-building corals. Warm western boundary currents can carry tropical water toward higher latitudes. The Kuroshio Current supports coral communities extending northward near Japan. The East Australian Current carries warm water southward along Australia. The Gulf Stream influences western Atlantic climates, although true reef development becomes limited at higher latitudes. Temperature alone does not determine reef presence. Light aragonite saturation, winter extremes, substrate, and competition also matter. Some subtropical reefs contain fewer species but may become increasingly important as ranges shift. Poleward coral expansion has been observed in some regions. Expansion is not guaranteed. Suitable habitat, larval supply, and winter survival are required. Tropical species entering temperate habitats interact with established kelp, algae, fishes, and invertebrates. Warming can reduce kelp and favor tropical herbivores in some transitional zones. This process is sometimes called tropicalization. Reef water chemistry reflects metabolism. Photosynthesis consumes carbon dioxide and produces oxygen. Respiration consumes oxygen and produces carbon dioxide. Calcification removes calcium and carbonate from dissolved form. Calcium carbonate dissolution returns them to the water. These processes change dissolved inorganic carbon and total alkalinity. Dissolved inorganic carbon includes carbon dioxide, bicarbonate, and carbonate. At normal seawater pH, bicarbonate is the dominant form. Total alkalinity measures the acid-neutralizing capacity of water. It is influenced mainly by bicarbonate, carbonate, and borate, with smaller contributions from other compounds. Calcification reduces total alkalinity by approximately 2 units for each unit of calcium carbonate formed on a molar basis. Photosynthesis changes dissolved inorganic carbon but has little direct effect on total alkalinity. Measurements of oxygen, pH, dissolved inorganic carbon, and alkalinity can estimate community metabolism. Researchers place sensors over reefs or collect water moving across them. They calculate net community production and net community calcification. Net community production is photosynthesis minus respiration. Net community calcification is calcification minus dissolution. A reef may be net productive during the day and net respiratory at night. Over a full day, the balance may be positive, negative, or near zero. Season, tides, cloud cover, temperature, and community composition alter the result. High algal production does not necessarily indicate a healthy coral reef. A degraded reef may have high photosynthesis from algae, but low structural growth. The physical environment also controls gas exchange at the sea surface. Waves increase exchange of oxygen and carbon dioxide with the atmosphere. Warm water holds less dissolved oxygen than cool water. Oxygen solubility also decreases with increasing salinity. Hypoxia means unusually low oxygen. Anoxia means no measurable oxygen. Open reef slopes are usually well oxygenated because of waves and currents. Protected lagoons can become hypoxic when respiration is high and water exchange is low. Nutrient enrichment can stimulate plankton and algae. Their decomposition consumes oxygen. Mass mortality may occur if oxygen falls below the tolerance of fishes and invertebrates. Corals can survive brief low oxygen periods in some habitats. Prolonged hypoxia damages tissue and disrupts metabolism. Heat increases respiration while decreasing oxygen solubility. This combination raises stress. Coral metabolism is influenced by body size and colony shape. Small colonies have high surface area relative to volume. They exchange materials efficiently but are easily overgrown. Large colonies store more energy and reproduce more, but parts of the colony may experience different flow and light. Branch tips often grow faster than shaded bases. Growth zones within one colony can differ genetically only through rare mutations, but they differ physiologically because of microenvironment. Somatic mutations occur as cells divide. A large old colony may contain several genetic cell lineages. Some mutations can be passed into gametes if reproductive tissue develops from the affected lineage. The evolutionary significance of somatic variation in corals is still being studied. Coral energy is stored as lipids, proteins, and carbohydrates. Lipid reserves are especially important during bleaching, reproduction, and low food periods. Colonies with high energy reserves may survive longer without symbiont-derived carbon. Heterotrophic feeding can replenish reserves. Not all species can increase feeding equally. Polyp size, prey availability, water flow, and behavior matter. Corals capture zooplankton, but they also consume bacterioplankton and particulate organic matter. Some absorb dissolved free amino acids. Nitrogen waste is often released as ammonium. Symbiotic algae rapidly take it up. This tight recycling reduces nutrient loss. Phosphorus is recycled to organic compounds and phosphate. The ratio of carbon, nitrogen, and phosphorus influences growth. The Redfield ratio is a broad average for marine plankton. It is about 106 atoms of carbon to 16 atoms of nitrogen to 1 atom of phosphorus. Coral tissues and symbionts do not always follow this ratio. Their elemental composition changes with feeding, light, reproduction, and nutrient supply. Carbon-rich but nutrient-poor conditions can limit tissue growth while allowing energy storage. Nutrient-rich conditions can increase symbiont density, sometimes making corals appear darker. Very dense symbiont populations can increase self-shading and oxygen stress. The coral host controls symbiont numbers through nutrient limitation, digestion, expulsion, and cell regulation. Symbionts divide inside host tissues. Their population is not static. During bleaching, symbiont density can fall dramatically. Recovery requires cell division, acquisition, or both. Temperature affects enzyme activity. Each enzyme has an operating range. At high temperature, proteins lose structure, membranes become unstable, and metabolic reactions become imbalanced. Heat shock proteins help stabilize and repair damaged proteins. Antioxidant enzymes neutralize reactive oxygen species. Superoxide dismutase converts superoxide radicals into hydrogen peroxide. Catalase and peroxidases then help break down hydrogen peroxide. If reactive oxygen production exceeds antioxidant stress develops, mitochondria and chloroplasts are major sources of reactive oxygen. Symbiodiniaceae chloroplasts are especially sensitive when light energy cannot be processed normally. The photosynthetic system contains two main reaction centers, called photosystem I and photosystem II. Heat can damage the proteins associated with photosystem II. When electron transport is disrupted, excess electrons react with oxygen. Corals can reduce light exposure by retracting tissue, changing pigments, or moving symbionts within cells. They can also dissipate excess energy as heat through non-photochemical quenching. Symbiont types differ in these protective mechanisms. Photoinhibition occurs when light reduces photosynthetic efficiency. Temporary photoinhibition is reversible. Chronic photoinhibition is reversible. Chronic photoinhibition causes longer-lasting damage. Researchers measure photosynthetic efficiency with pulse amplitude modulated fluorometry. This technique examines chlorophyll fluorescence. A decline in maximum quantum yield can indicate stress before visible bleaching occurs. Fluorometry is useful but not sufficient by itself. Values vary naturally with time of day, species, and light history. Coral respiration can be measured in sealed chambers. Oxygen change is recorded in darkness. Photosynthesis is estimated from oxygen production under light. Chambers alter flow and chemistry, so experiments must be designed carefully. Field measurements provide realism but are harder to control. Laboratory aquariums allow precise temperature, light, and pH treatments. Results from aquariums may not fully represent the complexity of natural reefs. Strong studies combine laboratory experiments, field observations, and long-term monitoring. Coral growth is measured in several ways. Buoyant weighing estimates skeletal mass by comparing weight in water. The alkalinity anomaly technique calculates calcification from changes in alkalinity. Calcine and other fluorescent dyes mark the skeleton at a known time. Later growth appears beyond the fluorescent band. Computed tomography scans reveal internal density and growth bands without cutting the whole colony. Three-dimensional imaging measures volume and surface area. Linear extension describes how far the colony extends. Skeletal density describes mass per unit volume. Calcification is the product of extension and density. A coral may maintain extension by producing a less dense skeleton. This can make growth appear normal while structural strength declines. Temperature often increases calcification up to an optimum. Above the optimum, calcification falls. The optimum differs among species and populations. Light generally increases growth until photosynthesis becomes saturated or photoinhibition begins. Food can support growth even under low light. Sediment and pollutants reduce growth by increasing cleaning and repair costs. Coral reproduction also follows energetic cycles. Egg production requires large lipid investment. Some colonies reduce growth before spawning as energy moves into gametes. After spawning, tissue energy reserves may be low. This can affect stress tolerance. Reproductive timing is influenced by seasonal temperature cycles. In regions with weak seasonality, species may spawn at different times of year. Lunar cues help synchronize the final release. Corals detect light through photoreceptor proteins. Cryptochromes and auxins are among the proteins studied. Corals do not have eyes, but they respond to light direction, color, intensity, and daily timing. They also possess genes associated with circadian rhythms. A circadian rhythm is an internal cycle lasting about 24 hours. It continues under constant conditions, but is adjusted by environmental signals. Coral gene expression changes between day and night. Photosynthesis-related stress responses peak during light periods. Feeding, calcification, and cell repair may follow daily patterns. Seasonal rhythms affect reproduction and growth. The nervous system of a coral is a diffuse nerve net. There is no centralized brain. Nerve cells coordinate tentacle movement, contraction, feeding, and responses to touch or chemicals. Colonial integration varies. A stimulus to one polyp can sometimes produce responses across part of the colony. Electrical signals and chemical messengers travel through connected tissues. Corals use neurotransmitter-like substances also found in other animals. Their simple organization does not mean their physiology is simple. They maintain ion gradients, recognize self and non-self, control symbionts, repair wounds, and coordinate reproduction. Self-recognition matters when colonies meet. Fragments from the same genetic individual may fuse. Closely related colonies may sometimes fuse during early life. Unrelated colonies usually form boundaries and attack one another. Fusion between incompatible colonies can lead to tissue rejection. Histocompatibility systems reduce the risk of foreign cells spreading through the colony. Chimerism occurs when genetically different individuals fuse and persist. Chimerate colonies may contain several genotypes. This can increase genetic diversity within one structure, but it can also create internal conflict. The frequency and ecological importance of chimerism differ among species. Coral skeletons host endolithic organisms. Endolithic algae live inside microscopic spaces in the carbonate. The green alga Ostrobium is common within coral skeletons. It survives under extremely low light by using wavelengths that penetrate tissue and skeleton. During bleaching, light enters the skeleton more easily. Endolithic algae may photosynthesize more. They can transfer some organic carbon to coral tissue, although the importance of this support varies. Endolithic microbes also contribute to bioerosion. They dissolve carbonate to create space. The skeleton, therefore, remains biologically active even below the living tissue. Boring sponges chemically dissolve and mechanically remove carbonate. They produce small chips that become sediment. Sponge bioerosion often increases in nutrient-enriched water. Higher carbon dioxide can also increase chemical dissolution by some bioeroders. This creates another pathway by which environmental change can weaken reefs. Crostose coralline algae help cement loose material. They deposit high magnesium calcite, a mineral that can be sensitive to acidification. Coralline algae also experience bleaching under heat stress. Loss of coralline algae reduces settlement cues and surface stability. Reef cementation includes both biological and inorganic processes. Minerals precipitate in cavities where water chemistry favors deposition. Microbial activity changes local alkalinity and pH. Cements bind grains and fragments into solid limestone. The internal reef framework contains open cavities. These cavities may make up a large share of total volume. They provide habitat for cryptic organisms and channels for water flow. Sediment gradually fills some cavities. Cement strengthens others. The reef is therefore not a solid block. It is a porous, changing structure. Pressure from accumulating material can compact deeper layers. Chemical alteration changes original aragonite into calcite in some settings. Freshwater exposure can dissolve minerals and create secondary deposits. Geologists call these post-depositional changes diagenesis. Diagenesis can obscure the original biological structure. It can also preserve chemical records or create new ones. Interpreting ancient reefs requires separating original signals from later alteration. Fossil reefs provide evidence about past sea levels. Because reef-building corals live near the surface, their fossil positions can indicate former ocean height. Tectonic uplift may raise old reefs above modern sea level. Subsidence may lower them below it. Dating uplifted coral terraces helps estimate land movement and sea level history. Coral terraces occur in places such as Barbados, Papua New Guinea, and islands of the Pacific. They record repeated glacial and interglacial cycles. During warm interglacial periods, sea level rose. During glacial periods, sea level fell as ice sheets expanded. Reefs repeatedly grew, were exposed, eroded, and flooded. The present interglacial period began about 11,700 years ago. Many modern reefs reached near their current sea level several thousand years ago. The exact timing varied regionally because land and ocean basins continued adjusting after ice loss. This adjustment is called glacial isostatic adjustment. The weight of ice depressed continental crust. After melting, the crust slowly rebounded. Water redistribution also changed Earth's gravitational field and rotation. Sea level history is therefore not identical at every location. Local reef growth also depends on subsidence, uplift, sediment supply, and currents. Darwin's basic subsidence sequence remains useful, but modern reef geology includes these additional processes. Atolls often contain freshwater lenses beneath their islands. Rainwater infiltrates porous sediment and floats above denser seawater. The freshwater lens is thin and vulnerable to drought, contamination, and saltwater intrusion. Island vegetation stabilizes sediment and adds organic material. Roots trap particles. Leaves contribute soil. Seabirds, marine organisms, and human activity add nutrients. Atoll islands are not simply fixed piles of coral sand. They are dynamic landforms shaped by biology and waves. Some reef islands have increased in area despite sea level rise because sediment continues to accumulate. Others have eroded or become less habitable. Area alone does not determine safety. Flooding frequency, freshwater quality, shoreline position, infrastructure, and storm exposure also matter. Now let us continue toward reef biogeography and evolution. Reef species distributions are shaped by plate tectonics. Continents and ocean basins move over millions of years. The tropical Tethys Ocean once supported extensive reefs between ancient land masses. As continents shifted, connections narrowed and closed. The modern Indo-Pacific and Atlantic reef faunas became separated. The Indo-Australian archipelago contains many islands, shallow seas, and complex current systems. This region maintained habitat through changing sea levels and tectonic movement. Its environmental complexity likely contributed to high species formation and persistence. The coral triangle is sometimes described as both a center of origin and a center of overlap. Some lineages may have evolved there. Others arrived from surrounding regions and accumulated. Scientific debate continues over the relative importance of these mechanisms. Species diversity generally declines eastward across the Pacific and westward across the Indian Ocean. Isolation limits larval exchange. Remote islands may contain fewer species but high levels of endemism. An endemic species occurs naturally only in a particular region. Hawaiian reefs contain many endemic fishes. Hawaii is extremely isolated, more than several thousand kilometers from major continental reef systems. Its coral diversity is lower than that of the Western Pacific. Its fish fauna includes unique evolutionary lineages. The Eastern Tropical Pacific also has relatively low coral diversity. Strong climatic variability, upwelling, and historical isolation influenced this region. The Isthmus of Panama separates it from the Caribbean. Some related species occur on opposite sides because their ancestors were separated when the seaway closed. This is called vicariance. The Red Sea has high coral diversity and unusual environmental conditions. Its northern portion experiences strong salinity and seasonal temperature variation. Some northern Red Sea corals tolerate temperatures higher than their local summer maximum in experiments. This apparent thermal tolerance has attracted research interest. However, these corals remain vulnerable to pollution, coastal development, and future warming. Their heat tolerance does not make them invulnerable. The Persian Gulf contains reefs adapted to extreme seasonal temperatures. Summer water can exceed 34 degrees Celsius. Winter temperatures can fall below 20 degrees. Coral diversity is lower than in the open Indian Ocean. Many colonies experience frequent bleaching and high mortality. Extreme tolerance may involve particular genotypes, symbionts, and acclimatization. The Gulf also has high salinity and strong human pressure. The Great Barrier Reef extends for more than 2,000 kilometers along northeastern Australia. It is not one continuous reef. It contains thousands of individual reefs and hundreds of islands. Inner shelf reefs experience more land influence. Outer shelf reefs receive clearer oceanic water. Conditions vary greatly from north to south and from coast to open sea. Management therefore requires regional rather than uniform approaches. The Mesoamerican Reef extends along the coasts of Mexico, Belize, Guatemala, and Honduras. It is the largest barrier reef system in the western Atlantic. It supports fisheries, tourism, coastal protection, and diverse habitats. Caribbean reefs historically contained abundant branching Acropora palmata and Acropora cervicornis. These species built complex, shallow water structures. Disease, storms, warming, and local stress caused major declines. Some reefs shifted toward dominance by algae, sponges, or stress-tolerant corals. Restoration programs now grow Acropora fragments in nurseries and transplant them to reefs. The Florida Reef Tract extends along the Florida Keys. It has experienced repeated bleaching, disease, hurricanes, cold events, and poor water quality. Stony coral tissue loss disease caused severe mortality beginning in the 2010s. Active interventions include disease treatment, land-based nurseries, genetic banking, and assisted reproduction. Brazilian reefs occur in turbid western Atlantic water. They contain distinctive species and extensive rhodolith beds. Rhodoliths are unattached nodules formed mainly by coralline algae. They create habitat on the seafloor. West African coral communities are more limited because of cool currents, river sediment, and upwelling. The eastern Atlantic has fewer reef-building coral species than the Caribbean. The western Indian Ocean includes reefs around East Africa, Madagascar, the Seychelles, the Maldives, and island groups. The 1998 global bleaching event caused severe mortality in parts of this region. Recovery varied according to depth, currents, herbivory, and later disturbance. The Maldives consist of atolls built on submerged volcanic foundations. Channels between atolls allow strong current flow. Reef islands support dense human settlements and tourism. Their low elevation creates sensitivity to flooding and coastal change. The Seychelles include granitic islands and coral atolls. Some reefs suffered very high mortality in 1998. Certain sites later recovered through coral recruitment. Others shifted toward persistent macroalgal dominance. Recovery was more likely where herbivorous fish biomass remained high and structural conditions were suitable. The Chagos Archipelago contains remote reefs in the central Indian Ocean. Low local population pressure reduces fishing and coastal pollution. However, remote reefs still experience marine heat waves and bleaching. This demonstrates that global stress reaches sites far from direct local disturbance. Local protection cannot stop ocean warming. It can reduce additional pressure and improve recovery potential. Pacific reefs include vast numbers of islands and atolls across Melanesia, Micronesia, and Polynesia. Many communities depend directly on reef fisheries. Traditional management systems include seasonal closures, customary tenure, restricted areas, and cultural rules. These systems vary widely. Some align closely with modern conservation goals. Others were designed for social or ceremonial reasons. Successful management often combines local knowledge, clear rights, scientific monitoring, and community enforcement. Reef organisms have dispersed across the Pacific over millions of years. Ocean currents create stepping stone connections among islands. Rare long-distance dispersal events can establish new populations. After establishment, isolation can produce genetic divergence. Speciation occurs when populations become reproductively isolated. Isolation may be geographic, ecological, behavioral, or genetic. Coral species can also hybridize. Hybridization occurs when distinct species reproduce and produce offspring. Some coral hybrids are fertile. Hybridization may transfer useful genes between species that can also blur taxonomic boundaries. The Caribbean elkhorn and staghorn corals can hybridize, producing Acropora prolifera. The hybrid is common in some locations. Historically, it was considered less important for reef building. Its abundance may increase where parent species decline. Polyploidy, incomplete lineage sorting, and reticulate evolution complicate coral genetics. Incomplete lineage sorting occurs when ancestral genetic variants persist through several speciation events. Different genes may therefore suggest different evolutionary relationships. Reticulate evolution describes networks of genetic exchange rather than a simple branching tree. Coral taxonomy increasingly uses whole genome data to resolve these patterns. A coral genome contains the genetic material of the animal host. Symbionts and microbes have separate genomes. When researchers sequence a coral sample, they must distinguish among these sources. Reference genomes exist for several coral species. Genomic studies identified genes related to heat tolerance, immunity, calcification, reproduction, and symbiosis. Gene expression studies measure ribonucleic acid. They show which genes become more or less active under particular conditions. Epigenetic processes modify gene activity without changing the underlying deoxyribonucleic acid sequence. Examples include deoxyribonucleic acid methylation and modifications to histone proteins. Epigenetic changes may contribute to acclimatization. Some may persist across cell divisions or generations. Evidence for stable transgenerational epigenetic inheritance in corals is growing but remains incomplete. Parental environment can affect offspring condition through egg nutrients, symbionts, microbes, and molecular signals. These effects can improve or reduce offspring performance. Larval experiments test survival under heat, low pH, sediment, and pollutants. Juvenile corals often respond differently from adults. A condition tolerated by an adult colony may prevent larval settlement. Population models must include all life stages. Adult survival alone is not enough if recruitment fails. Likewise, high larval production is not enough if adults repeatedly die. Coral demography tracks colony birth, growth, shrinkage, fusion, fragmentation, and death. Colonies can partially die and later regrow. This makes coral demography more complicated than counting separate individuals. Researchers map individual colonies over time. They measure area, tissue loss, and reproductive size. Matrix models divide colonies into size classes. Integral projection models treat size as a continuous variable. These models estimate population growth rate and sensitivity to different life stages. For long-lived massive corals, adult survival may have the greatest effect. For fast-growing branching species, recruitment and fragmentation may be more important. Disturbance changes these relationships. A cyclone can remove large colonies and create fragments. A disease may selectively kill certain species. Bleaching can reduce growth across many size classes. Population recovery therefore depends on the type of disturbance. Ecological succession describes changes in community composition through time. After a severe disturbance, bare skeleton is colonized by microbes and algae. Grazers may reduce algae. Coral larvae settle. Fast-growing species can dominate early recovery. Later, slower-growing massive corals may increase. This sequence is not fixed. If algae remain dense, coral recruitment may stay low. If unstable rubble persists, young colonies may be crushed. If repeated heat waves occur, sensitive species may never regain dominance. A reef can shift into an alternative state. Macroalgal dominance is one possible state. Sponge dominance, soft coral dominance, rubble fields and low-complexity coral communities are others. The term phase shift is used when the change is substantial and persistent. Not every increase in algae is a permanent phase shift. Seasonal algae can decline naturally. Long-term data are needed to distinguish temporary fluctuation from stable change. Feedbacks can maintain a degraded state. Macroalgae trap sediment and reduce coral settlement. Low coral cover reduces structural habitat for herbivorous fish. Lower grazing allows more algae. Rubble movement kills recruits, preventing framework recovery. These feedbacks make restoration difficult. Positive feedbacks can also support recovery. Coral structure shelters fishes. Grazers keep settlement surfaces open. New corals add more structure. Coralline algae stabilize the substrate. Management attempts to strengthen these recovery processes. Now we will continue toward human use, environmental pressures, and the methods being developed to protect and rebuild reefs. Management begins with understanding how people use reefs. Coral reefs support food production, employment, trade, recreation, cultural identity, shoreline protection, and scientific research. Hundreds of millions of people live close enough to coral reefs to receive direct or indirect benefits from them. The exact number depends on how distance and dependence are defined. Some households rely on reef fish for protein. Others earn income through fishing, tourism, transport, construction, aquaculture, or the sale of marine products. Reef fisheries are usually multi-species fisheries. Fishers may catch dozens or hundreds of species using different gears. Common methods include hand lines, traps, nets, spears, and collection by hand. Small-scale fisheries can provide food and income with relatively little industrial equipment. They're also difficult to monitor because catches are distributed among many landing sites. Official statistics may record only part of the total harvest. Fish consumed within households or sold informally may never enter national records. The sustainability of a reef fishery depends on fishing effort, gear type, species biology, habitat condition, and management. Fast-growing small fishes can often tolerate more harvest than slow-growing large predators. Species that form predictable spawning aggregations are especially vulnerable. Fishers can remove a large share of the breeding population during a short seasonal event. Protection during spawning periods can reduce this risk. Size limits are another management method. Minimum size limits allow young fish to reproduce before capture. Maximum size limits can protect large adults with high reproductive output. A slot limit permits capture only within a middle size range. The best rule depends on the reproductive biology of the species. For sex-changing fishes, protecting large individuals can be particularly important. Gear restrictions can reduce habitat damage and unwanted catch. Fine mesh nets capture juveniles and small species. Heavy traps may break corals when placed or retrieved. Lost traps can continue catching animals. This is called ghost fishing. Biodegradable escape panels allow lost traps to open after a period of time. Spearfishing is selective when fishers can identify species and size before capture. It can still remove large individuals very efficiently. Night spearfishing can be especially effective because many fishes rest in predictable shelters. Explosives and poisons cause severe damage and are illegal in many countries. Blast fishing kills or injures fishes with pressure waves. It also shatters the reef framework. Repeated explosions produce unstable rubble fields that may remain unsuitable for coral recovery for decades. Cyanide has been used in some live fish trades to stun reef fishes. It can injure target fishes, kill non-target organisms, and damage coral tissue. Detection and enforcement can be difficult. Destructive methods often persist where poverty, weak governance, market demand, or conflict limits alternatives. Enforcement alone is rarely sufficient. Management works better when communities have secure access rights, practical alternatives, and confidence that rules apply fairly. Marine protected areas are widely used in reef conservation. A marine protected area is a defined marine region managed for conservation or resource objectives. Protection levels vary greatly. Some areas prohibit all extraction. Others allow fishing, tourism, transport, or traditional use under specified rules. A no-take zone prohibits removal of animals, plants, and minerals. Well-enforced no-take zones often contain greater fish biomass and larger individuals than nearby fished areas. Effects on coral cover are less consistent because corals are strongly influenced by heat, storms, disease, and water quality. Protected herbivores can reduce macroalgae and support recovery under suitable conditions. Protected predators can restore more natural size and age structures. Benefits can extend beyond boundaries. Adult fishes may move into surrounding fishing grounds. Larvae produced inside protected areas may disperse to other reefs. This export is called spillover when it involves the movement of older animals, and larval subsidy when it involves offspring. The amount of export depends on reserve size, habitat, currents, species movement, and fishing pressure outside. Very small protected areas may not contain the full home range of mobile species. Very large areas can be difficult to enforce. Networks of protected areas can represent different habitats and spread risk. A network may include warm reefs, cooler reefs, nurseries, spawning grounds, and migration corridors. Connectivity among sites helps populations exchange larvae. Replication ensures that one disturbance does not affect every protected example of a habitat. Climate planning increasingly considers future heat exposure. Some reefs regularly receive cooler water from upwelling or internal waves. Others experience strong currents, cloud cover, or natural temperature variability. These features may reduce stress in some years. They do not provide permanent protection from continued warming. Protected area effectiveness depends heavily on governance. A boundary on a map does not protect a reef unless rules are understood, accepted, funded, and enforced. Local participation can improve compliance. People are more likely to support measures when decision-making is transparent and benefits are visible. Monitoring is needed to determine whether management is working. Biological indicators include fish biomass, coral recruitment, coral cover, species composition, and disease. Social indicators include income, food security, compliance, conflict, and public trust. A policy can improve fish abundance while creating unfair costs for particular households. Conservation planning must therefore include both ecological and social effects. Tourism is another major reef use. Visitors pay for diving, snorkeling, boating, accommodation, food, and transport. Tourism revenue can create incentives to maintain healthy reefs. It can also finance protected areas and local employment. Poorly managed tourism causes physical damage, pollution, crowding, and wildlife disturbance. Divers and snorkelers may touch or kick corals. Boat anchors can break colonies and drag across the seafloor. Permanent mooring buoys reduce the need for anchoring at popular sites. Tour operators can limit group size and provide instructions before entering the water. Well-trained guides reduce contact and improve visitor behavior. Coastal resorts may increase sewage, freshwater demand, artificial light, and construction. Beaches are sometimes modified or replenished with imported sediment. Dredging for harbors and channels suspends fine particles. Environmental impact assessments attempt to predict these effects before construction begins. Their quality varies. A strong assessment considers currents, seasonal spawning, sediment transport, cumulative development, and realistic accident scenarios. Monitoring during construction allows work to stop if sediment limits are exceeded. Mitigation should not be treated as a substitute for avoiding unnecessary damage. Moving corals away from a construction site can save some colonies. Translocation usually causes stress, partial mortality, and loss of ecological context. Large old colonies are difficult to move successfully. Avoiding the impact is generally more reliable than relocating the organisms afterward. Land-based pollution is one of the most manageable reef pressures. Wastewater contains nutrients, organic matter, pathogens, pharmaceuticals, detergents, and other chemicals. Untreated sewage can stimulate algae and microbial growth. Decomposition consumes oxygen. Human pathogens can affect swimmers, while marine pathogens may affect reef organisms. Improved sanitation benefits both public health and reefs. Wastewater treatment can remove solids, organic matter, nitrogen, phosphorus, and pathogens. The level of treatment required depends on discharge volume and local circulation. Even treated effluent can influence a poorly flushed lagoon. Submarine groundwater discharge is another pathway. Groundwater carrying fertilizers or sewage can enter the sea through porous rock and sediment. The source may be located far inland. Groundwater pollution can continue for years after surface practices improve because underground movement is slow. Tracing nutrients with isotopes can help identify their origin. Nitrogen from sewage may have a different isotopic pattern from nitrogen produced naturally in the ocean. Agriculture contributes fertilizers, soil, pesticides, and animal waste. Vegetated buffer zones slow runoff and trap sediment. Wetlands remove nutrients through plant uptake and microbial processes. Contour farming, reduced tillage, and careful fertilizer timing reduce losses. The most effective measures are adapted to local soil, rainfall, and crops. Urban runoff carries oil, metals, plastics, tire particles, and other contaminants. Storm drains can deliver this mixture directly to coastal water. Retention ponds and constructed wetlands hold water long enough for particles to settle. Green roofs, permeable pavement, and planted drainage systems reduce rapid runoff. Industrial pollution includes metals, petroleum compounds, solvents, heated water, and chemical waste. Copper is toxic to coral larvae at sufficiently high concentrations. Some herbicides interfere with photosynthesis in symbiotic algae. Oil coats surfaces and contains compounds that damage cells. Acute spills can kill organisms quickly. Chronic low-level exposure may reduce growth and reproduction without causing immediate visible mortality. Marine debris affects reefs at several scales. Large fishing nets become entangled on coral colonies. Waves move the nets repeatedly, breaking branches and abrading tissue. Fishing line wraps around gorgonians and sponges. Plastic bags and sheets cover organisms and reduce water exchange. Small plastic particles are ingested by many reef animals. Corals can capture microplastics with their tentacles or mucus. The long-term ecological consequences remain under study. Particles may occupy digestive space, carry chemicals, or alter microbial communities. Their effects depend on size, shape, polymer, concentration, and exposure time. Removing debris can provide immediate local benefit. Removal must be careful because pulling an entangled net can cause additional breakage. Divers often cut material into small sections and lift it gradually. Prevention is more efficient than repeated cleanup. Port reception facilities, fishing gear recovery programs, waste collection, and product design reduce inputs. Shipping creates additional risks. Ships can strike reefs or run aground. A grounding may crush thousands of square meters of structure. Fuel and cargo can be released. Salvage operations may cause further damage if vessels are dragged across the reef. Accurate charts, navigation systems, pilotage, and marked shipping lanes reduce risk. After a grounding, restoration teams may remove toxic material, stabilize rubble, and reattach surviving corals. Recovery from major physical injury is slow because the geological framework has been destroyed. Ships also transport non-native species in ballast water and on hulls, and introduced species may spread if environmental conditions are suitable and natural controls are absent. Lionfish provide a well-known example in the western Atlantic. Lionfish native to the Indo-Pacific became established in Atlantic and Caribbean reefs. They consume many small fishes and crustaceans. They mature quickly and reproduce frequently. Local predators have not controlled their population sufficiently. Targeted removal reduces lionfish abundance at selected sites. Regular removal is needed because surrounding populations replace captured individuals. Lionfish are edible when prepared safely, and fisheries have been encouraged in some regions. Commercial demand alone has not eliminated the invasion. Other reef invasions include algae, tunicates, corals, fishes, and pathogens. Prevention is generally less expensive than control after establishment. Biosecurity includes inspection, ballast water treatment, hull cleaning, and restrictions on aquarium release. The aquarium trade collects fishes, corals, mollusks, and other organisms. When managed well, it can provide income with relatively small harvest volumes. When poorly managed, it can deplete rare species and encourage destructive collection. Captive breeding reduces pressure for some fishes. Many clownfishes, gobies, cardinal fishes, and seahorses are bred commercially. Captive propagation of corals is also common. Not every species reproduces easily in captivity. Clear labeling helps buyers choose captive-bred organisms. International trade in stony corals is regulated under the Convention on International Trade in Endangered Species of Wild Fauna and Flora. Permits aim to ensure that trade is legal and not harmful to wild populations. Enforcement depends on accurate identification and reporting. Illegal wildlife trade can use false labels or hidden shipments. Climate change is the largest global threat to coral reefs. It affects temperature, sea level, storm behavior, rainfall, oxygen, and ocean chemistry. The increase in global temperature is driven mainly by greenhouse gases released through human activity. Carbon dioxide comes from fossil fuel use, cement production, and land use change. Methane, nitrous oxide, and other gases also contribute. The ocean absorbs most of the excess heat trapped in the climate system. Marine heat waves have become more frequent, longer, and more intense in many regions. A marine heat wave is a period of unusually high ocean temperature relative to local historical conditions. Its ecological effect depends on magnitude, duration, season, and previous exposure. Global bleaching can occur when heat stress affects several ocean basins during the same broad period. Such events have been documented repeatedly since the late 20th century. The first widely recognized global bleaching event occurred around 1998. Further global-scale events occurred during the 2010s and 2020s. Individual regions have also experienced severe events outside these global periods. The historical temperature baseline used to define anomalies is itself changing. A reef may acclimatize to some gradual warming, but the rate of extreme heat often exceeds that adjustment. Natural selection can increase thermal tolerance if tolerant colonies survive and reproduce. Adaptation is limited by genetic variation, generation time, population size, and the speed of environmental change. A population cannot adapt if mortality removes most breeders before successful reproduction. Larval exchange may introduce heat-tolerant genes from warmer reefs. It may also introduce genes poorly matched to local conditions. Gene flow can support or slow adaptation, depending on circumstances. Climate models project different futures according to greenhouse gas emissions. Lower emissions produce less warming and lower risk than high emissions. No emissions pathway avoids all coral loss. The difference between pathways remains extremely important. Every fraction of a degree affects the frequency and severity of heat stress. Reducing carbon dioxide emissions is therefore central to reef conservation. Local management cannot substitute for climate mitigation. It can reduce pollution, overfishing, and direct damage. This gives corals more energy and ecological support for recovery. Without stabilization of global temperature, local gains are repeatedly overwhelmed. Sea level rise has mixed direct effects on reefs. Slightly deeper water can reduce heat and light stress on very shallow reef flats. Greater depth can also reduce the light available for calcification. Healthy reefs can grow upward and produce sediment. Degraded reefs may not keep pace. Rising sea level increases wave energy reaching shore when reef crests become deeper. Coastal protection declines if structural growth does not match the rise. Stronger tropical cyclones are expected in some basins as the climate warms, although total cyclone frequency may not increase everywhere. Warm water provides more energy for intense storms. Storm tracks and rainfall patterns may also change. Corals have always experienced cyclones. The concern is the combination of storms with bleaching, disease, and reduced recovery intervals. A reef damaged by heat may be more easily broken by waves because dead skeleton weakens over time. Heavy rainfall creates low salinity and carries land pollutants. Flood plumes can spread far across shallow shells. Satellite images track their movement. Corals exposed to both heat and low salinity may experience greater mortality than under either stress alone. Climate change also reduces oxygen in the ocean. Warmer water holds less oxygen. Stronger stratification reduces mixing between surface and deep layers. Changes in productivity and respiration further alter oxygen concentration. Most shallow wave-exposed reefs remain oxygenated, but lagoons and coastal areas face greater hypoxia risk. Restoration has become a major field in reef science. Coral restoration aims to assist recovery through active intervention. It is not one technique. It includes coral gardening, larval propagation, substrate stabilization, disease treatment, predator control, cryopreservation, selective breeding, and habitat engineering. Coral gardening usually begins with small fragments. Fragments are grown in underwater or land-based nurseries. After reaching a suitable size, they are attached to degraded reefs. Branching corals are well suited to fragmentation because pieces grow rapidly. Fragments may be suspended on ropes, placed on frames, or attached to nursery trees. Nursery structures require cleaning and maintenance. Algae, predators, storms, disease, and equipment failure can cause losses. Growing many fragments from one colony produces genetically identical clones. Large numbers of clones increase coral cover but not genetic diversity. Restoration programs therefore track genotypes. A mixture of genotypes improves the chance that some colonies tolerate future conditions. Genetic diversity also supports successful sexual reproduction. Fragments of the same genotype can fuse when placed together. This process can create a large reproductive colony more quickly. It is sometimes called microfragmentation when very small pieces are used. Microfragmentation is especially useful for slow-growing massive corals. Small fragments are cut from a colony and placed close together. They grow rapidly along their edges. When they meet, genetically identical tissues fuse. The method increases surface coverage faster than waiting for one large fragment to expand. Cutting causes initial injury and requires careful handling. Fragments must be kept cool, shaded, and wet during processing. Tools and tanks must be disinfected to reduce disease transmission. Outplanting methods include cement, epoxy, nails, clips, cable ties, and specialized bases. Each method has costs and environmental effects. Attachment must resist waves without covering excessive living tissue. The substrate must be stable. Attaching a coral to loose rubble usually fails when the rubble moves. Restoration survival is often high during the first few months and lower over longer periods. Monitoring must continue for years. A project that reports only initial attachment success may overestimate its effect. Outplanted corals must eventually grow, reproduce, and contribute to reef structure. Scale is a major limitation. Nurseries can produce thousands or millions of fragments. Large reef systems contain billions of colonies in vast areas. Restoration is therefore most practical for selected sites, endangered populations, tourism areas, research, and recovery after localized damage that cannot replace climate action or broad ecosystem management. Sexual propagation increases genetic diversity. During spawning, researchers collect egg and sperm bundles. Gametes from different colonies are separated and mixed under controlled conditions. Fertilized embryos develop in tanks or floating enclosures. Larvae are offered settlement surfaces. The surfaces may be ceramic tiles, limestone pieces, or specially designed structures. Crustose coralline algae and microbial films improve settlement. Juveniles are raised until they are large enough for outplanting. Early mortality is high. Predators, algae, sediment, and competition kill many settlers. Producing very large numbers can compensate for low survival. Larval enhancement releases competent larvae directly onto reefs. Temporary mesh enclosures keep larvae over suitable substrate during settlement. The enclosure is removed afterward. This method can treat larger areas than placing individual fragments. Its long-term success depends on post-settlement conditions. If the reef remains covered with algae or unstable sediment, many juveniles die. Assisted gene flow moves individuals or gametes between populations. The aim is to increase genetic diversity or introduce stress-tolerant variants. Movement requires caution. A genotype adapted to warm water may be sensitive to cold. It may introduce disease or disrupt local genetic structure. Risk assessments examine environmental matching, disease screening, and reproductive compatibility. Selective breeding crosses parents with desirable traits. Researchers expose colonies or offspring to heat and identify tolerant families. Offspring from tolerant parents may show improved survival. The trait must remain effective under field conditions. Tolerance can involve trade-offs in growth, reproduction, or disease resistance. Breeding programs should therefore evaluate several traits rather than heat alone. Assisted evolution is a broad term for interventions intended to accelerate natural adaptive processes. It may include selective breeding, symbiont manipulation, acclimatization, microbiome treatment, and gene flow. The term does not necessarily mean genetic engineering. Genetic engineering directly changes genetic material using laboratory methods. Its use in coral conservation remains experimental and raises ecological, regulatory, and ethical questions. Any released organism could reproduce and spread beyond the original site. The benefits and risks require careful evaluation. Symbiont manipulation attempts to associate corals with more tolerant algal partners. Coral larvae or juveniles can be exposed to selected symbiont cultures. Some combinations improve heat survival. Host and symbiont compatibility is specific. A tolerant symbiont may not persist in the adult host. It may also reduce carbon transfer and growth. Microbiome manipulation uses beneficial microorganisms, sometimes called probiotics. Selected bacterial communities may improve nutrient cycling, pathogen resistance, or stress recovery. Experiments have shown promising effects in some species. The coral microbiome is complex and changes rapidly. Introduced strains may disappear after release. Safety assessment is needed because microbes can evolve and move among hosts. Preconditioning exposes corals to moderate stress before severe stress. Controlled heat pulses may induce protective responses. Nursery corals can also be grown under variable temperature or light. Preconditioned colonies sometimes survive later heat better. The protection may fade. Repeated conditioning can also consume energy and reduce growth. Cryopreservation stores biological material at very low temperature. Coral sperm can be frozen using protective chemicals and kept in genetic banks. After thawing, viable sperm may fertilize fresh eggs. Embryos and eggs are more difficult to preserve because they contain more water and complex structures. Methods for freezing larvae, tissues, and symbionts continue to improve. Cryobanks preserve genetic diversity even if wild colonies disappear. Stored material remains useful only if future reproduction and rearing techniques are available. Accurate records must connect each sample to species, genotype, location, and collection date. Living gene banks maintain coral colonies in aquariums or nurseries. They protect genotypes threatened by disease or habitat loss. Maintaining them is expensive and vulnerable to equipment failure. Backup populations in several facilities reduce risk. Disease treatment is another active intervention. Divers apply antibiotic paste to some bacterial disease lesions. Treatments can slow or stop tissue loss on individual colonies. They require repeated visits and do not remove the environmental cause. Large-scale antibiotic use raises concerns about resistance and effects on non-target microbes. Alternative treatments include antiseptics, probiotics, removal of diseased tissue, and physical barriers. No method is universally effective. Early detection improves the chance of control. Predator management focuses mainly on outbreaking crown-of-thorns sea stars and coral-eating snails. Divers may remove animals or inject them with approved solutions. Surveillance identifies outbreak fronts before density becomes extreme. Control is labor-intensive. It protects selected reefs rather than eliminating the species. Corallivorous snails in the genus Coralliophila can damage Caribbean corals. They may concentrate on nursery outplants. Manual removal can improve survival at small restoration sites. Algae are controlled through manual removal, herbivore protection, or experimental addition of grazers. Removing algae by hand creates temporary settlement space. Unless the cause of algal dominance changes, the algae return. Sea urchin restoration is being tested in the Caribbean. Hatcheries raise juvenile long-spined urchins for release. Young urchins require shelter and suitable food. Disease risk remains a concern. Releasing too many grazers can also increase bioerosion. The goal is a functional grazing community, not the maximum possible grazing rate. Substrate stabilization is essential on rubble fields. Loose fragments roll during waves and prevent coral settlement. Methods include mesh, rock structures, pins, cement, and biodegradable materials. Plastic mesh can become marine debris if it breaks loose. Natural fiber or mineral-based alternatives are being developed. Structures must match local hydrodynamics. A design that survives a sheltered lagoon may fail on an exposed crest. Artificial reefs provide hard structure. They can be made from concrete, stone, steel, ceramic, or other stable materials. Artificial reefs do not automatically create coral ecosystems. They may attract fish from nearby habitat without increasing total production. Material chemistry, shape, depth, stability, and placement influence colonization. Tires and unstable waste materials have performed poorly and caused damage at some historical projects. Purpose-built structures are more reliable. Complex surfaces provide shelter and attachment points. However, artificial complexity cannot fully reproduce the biological and geological functions of a mature reef. Electrochemical mineral accretion uses low electrical current to precipitate minerals on metal frames. Proponents suggest that corals grow rapidly on these structures. Results vary, and maintenance demands can be high. The electrical systems, cables, corrosion, and storm damage create practical challenges. More comparative research is needed to determine when the method offers advantages. Shading and cooling have been proposed for heat emergencies. Floating shade cloth reduce light stress over small areas. Pumping cooler water or increasing mixing may lower temperature locally. Marine cloud brightening has been proposed to increase cloud reflectivity over large reef regions. These methods remain experimental. They require forecasting, infrastructure, energy, and environmental assessment. A large reef cannot be covered easily. Emergency intervention may be valuable for nurseries, rare species, or small high-priority sites. It does not address the global cause of warming. Solar radiation modification at planetary scale has also been discussed. Such approaches would affect climate broadly and carry major uncertainties. They would not stop ocean acidification because atmospheric carbon dioxide would remain high. Coral conservation, therefore, remains closely connected to energy policy, land use, and greenhouse gas reduction. Environmental deoxyribonucleic acid and genomic monitoring may improve early warning. Heat-tolerant genotypes can be mapped. Pathogens may be detected before visible disease becomes widespread. Autonomous sensors can transmit temperature, oxygen, pH, and turbidity in real time. Forecast systems combine satellite data and ocean models. Managers can prepare nurseries, suspend construction, reduce local discharge, or deploy emergency teams before a predicted event. Forecasts cannot prevent heat waves. They can improve timing and reduce avoidable additional stress. Citizen science expands observation capacity. Divers, fishers, tourists, and coastal residents record bleaching, disease, debris, and species sightings. Standardized training improves data quality. Photographs allow experts to verify reports. Citizen data are especially useful in regions with limited scientific staff. Community observations also contain long-term knowledge not recorded in formal surveys. Fishers may recognize changes in spawning seasons, currents, and species abundance. Combining local knowledge with scientific measurements can reveal patterns missed by either source alone. Respectful collaboration requires consent, credit, and protection of culturally sensitive information. Education supports conservation when it explains practical cause and effect. General awareness does not always change behavior. People need access to alternatives, infrastructure, and fair rules. A fisher cannot follow a closure if there is no food or income replacement. A household cannot prevent sewage pollution without sanitation services. A tourist cannot use a mooring buoy if none has been installed. Effective conservation therefore connects knowledge with institutions and resources. Economic valuation estimates the benefits provided by reefs. These benefits include fisheries, tourism, shoreline protection, biodiversity, and cultural value. Monetary estimates can influence planning. They also have limitations. Not every cultural relationship can be represented by a price. A reef may be irreplaceable to a community even when market income is low. Cost-benefit analysis should not erase rights, identity, or ecological thresholds. Insurance programs have been developed for some reefs. Tourism businesses or governments pay premiums. After a qualifying storm, funds become available for rapid assessment and restoration. Early removal of debris and stabilization can improve recovery. The approach links coastal protection with financial planning. It remains limited to locations with suitable institutions and revenue. Legal rights for ecosystems have been proposed or adopted in some jurisdictions. These frameworks recognize reefs or other natural systems as entities with legal interests. Practical enforcement and representation vary. Traditional marine tenure can also give communities authority to regulate access. No single governance model fits every reef. International agreements influence reef conservation. The United Nations Framework Convention on Climate Change addresses greenhouse gases. The Convention on Biological Diversity addresses conservation and sustainable use. The Convention on International Trade in Endangered Species regulates trade in listed organisms. Regional seas agreements coordinate pollution and habitat management. Global commitments depend on national implementation. Reefs cross political boundaries through currents, migration, trade, and climate. Larvae produced in one country may settle in another. Pollution can move across borders. Shared monitoring and data improve regional planning. Scientific data must be accessible to be useful. Open databases contain temperature, bleaching, species, and monitoring records. Standardized formats allow comparison. Data gaps remain largest in remote and lower-income regions. Funding often arrives after a crisis and ends before long-term recovery can be measured. Stable monitoring programs provide greater value than short, disconnected projects. Reef science also depends on taxonomy, museum collections, and basic natural history. A preserved specimen collected decades ago may reveal a species' former range. Genetic analysis can identify historical diversity. Old skeletal samples contain climate records. Maintaining collections requires careful storage and documentation. Training new taxonomists is equally important. Many reef species remain unnamed. Without identification, decline can occur before a species is recognized. Small cryptic organisms may perform major functions in decomposition, grazing, or nutrient cycling. The loss of these species is difficult to detect through coral cover surveys. Future monitoring will likely combine direct observation, genetics, imaging, acoustics, chemistry, and automated instruments. No method replaces all others. Satellite data provides scale, divers provide detail. Genomics provides biological mechanism. Long-term local records provide history. Models integrate these sources and explore possible futures. Models are simplified representations. Their predictions depend on assumptions and data quality. Uncertainty should be stated rather than hidden. A range of outcomes is often more informative than one precise number. Climate models project ocean conditions. Ecological models translate those conditions into coral growth, mortality, and recovery. Socioeconomic models examine fishing, tourism, and management. Integrated models attempt to connect all three. Complexity does not guarantee accuracy. Simple models can be useful when mechanisms are clear. Validation against observed data is essential. Experimental reefs and large aquarium systems allow controlled ecosystem studies. Researchers manipulate temperature, carbon dioxide, nutrients, and species composition. Such systems reveal interactions that are difficult to isolate in the field. They remain smaller and simpler than natural reefs. Mesocosms occupy an intermediate scale between laboratory tanks and open ecosystems. They contain natural water, sediments, and communities under partly controlled conditions. Field transplantation experiments compare coral performance across environments. Fragments from one colony are placed at several sites. This separates genetic effects from environmental effects. Reciprocal transplantation moves colonies in both directions between locations. A coral may perform best at its home site, suggesting local adaptation. It may perform equally well elsewhere, suggesting broad tolerance. Transplantation itself causes stress and must be included in the design. Common garden experiments raise different genotypes under identical conditions. Performance differences are then more likely to reflect genetics or inherited effects. Breeding designs estimate heritability. Heritability describes how much variation in a trait is associated with genetic differences in a particular population and environment. It does not mean the trait is fixed. Heritability can coexist with strong environmental influence. Thermal tolerance is not one trait. It includes cellular protection, symbiont function, energy storage, immunity, feeding, and recovery. Different mechanisms may produce similar survival during one experiment. A colony tolerant of short, intense heat may not tolerate a long, moderate event. Standardized tests improve comparison, but natural heat waves remain variable. The future of reefs will not be uniform. Some reefs are likely to lose most framework-building corals. Some will retain reduced but functioning communities. Some may recover after severe disturbance. Others may shift toward different coral species. High-latitude regions may gain tropical species while equatorial regions lose them. Novel reef communities will contain combinations not common in the past. A future reef may have lower diversity, smaller colonies, and less structural complexity. It may still provide habitat and coastal protection. Conservation must decide whether to preserve historical composition, maintain ecological function, protect particular species, or support adaptation. These goals can conflict. Restoring one fast-growing species may increase cover quickly but reduce diversity. Protecting a remote reef may conserve biodiversity, but provide fewer direct social benefits than restoring an urban reef. There is no purely technical answer. Decisions involve values, risk, cost, and fairness. Tierage is the prioritization of limited resources. The term is controversial in conservation because it can imply abandoning some reefs. In practice, every program already makes choices about sites and species. Transparent criteria make those choices easier to evaluate. Criteria may include biodiversity, climate exposure, connectivity, cultural importance, feasibility, and local support. Protecting climate refuges is one strategy. A refuge is an area expected to experience less severe climate stress or better recovery conditions. Refuges can be temporary. A location protected by currents today may warm in later decades. Refuge planning should therefore include networks and future projections. Evolutionary refugia preserve genetic diversity and adaptive potential. A population living under extreme natural conditions may contain useful traits. It should not be treated merely as a source of genes. The ecosystem and community around it also deserve protection. Coral reefs have survived major environmental changes over geological time. Individual reef systems and species have also disappeared. Survival of the broad coral lineage does not guarantee survival of modern reefs or the societies that depend on them. Past changes often occurred over much longer periods than current warming. Species could shift ranges as climate zones move. Today, coastlines, pollution, habitat fragmentation, and rapid temperature increase restrict those responses. The fossil record contains reef gaps lasting millions of years after mass extinctions. Reef building eventually returned, but on timescales irrelevant to human communities. This distinction is important. Geological survival is not the same as ecological continuity. The loss of coral cover can occur within weeks. The loss of framework may take years. The rebuilding of a mature reef may require decades or centuries. A reef several meters thick represents the accumulated work of many generations. Its living surface is only the current stage of that process. Coral reefs are sometimes described mainly through crisis. They are also systems of ordinary biological activity. Every day, polyps feed and respire. Algae fix carbon. Fishes graze, sponges filter water, microbes recycle nutrients. Larvae settle, skeletons grow by thin increments. Erosion removes part of that growth. The reef persists through the balance of these small processes. Scientific understanding has moved from viewing corals as simple animals to viewing them as partnerships of animals, algae, microbes, and minerals. The reef itself is now understood as a coupled biological, chemical, physical, and social system. Changes in one part spread into others. Heat alters symbiosis. Symbiosis alters calcification. Calcification alters habitat. Habitat alters fish communities. Fish communities alter algae. Human decisions alter every stage. This complexity makes prediction difficult. It also creates several points where management can help. Wastewater can be treated. Fishing can be regulated. Anchors can be replaced with moorings. Diseased colonies can sometimes be treated. Genetic diversity can be stored. Larvae can be raised. Greenhouse gas emissions can be reduced. None of these actions is sufficient alone. Together, they determine how much reef function remains. The most effective strategy is usually layered. Global emissions reduction limits future warming and acidification. Regional planning protects connectivity and water quality. Local management controls fishing and direct damage. Restoration supports particular populations and sites. Research improves each layer. Monitoring shows whether the combined effort is working. Success should be measured over appropriate time scales. A nursery can be built in months. A transplanted colony may mature in years. A reef framework develops over decades. Carbon dioxide remains in the climate system for much longer. Policies must therefore continue beyond short funding cycles. Long-term stewardship is less visible than emergency response, but it is more important. Maintenance includes checking moorings, enforcing rules, sampling water, recording disease, and supporting local institutions. These routine tasks rarely produce dramatic results. They prevent gradual failure. The science of reefs remains incomplete. Researchers continue to discover new species, genes, symbioses, diseases, and chemical pathways. The response of many deep and remote reefs is poorly known. The capacity for adaptation is uncertain. The practical scale of restoration is uncertain. The future rate of emissions is determined by human choices and is therefore also uncertain. Uncertainty does not mean nothing is known. Heat causes bleaching. Carbon dioxide changes ocean chemistry. Pollution and overfishing reduce resilience. Healthy, diverse populations recover more effectively than depleted ones. Early action preserves more options than delayed action. These conclusions are supported by observation, experiment, and physical chemistry. Coral reefs are among the most biologically dense environments in the ocean. Their productivity is maintained through close recycling in water that is often poor in nutrients. Their structure is built by organisms, but shaped continuously by waves, dissolution, erosion, and sediment. Their diversity depends on millions of interactions occurring across microscopic and regional scales. A coral polyp may be only a few millimeters wide. Its skeleton can contribute to a reef visible from space. A larva may drift for days before selecting a surface. Its colony may persist for centuries. A heat anomaly of only a few degrees above the local seasonal maximum can disrupt this entire partnership. The sensitivity of reefs comes from their dependence on stable relationships. Their persistence comes from reproduction, variation, connectivity, and repeated repair. The complete science of coral reefs, therefore, has no single central subject. It begins with the cell and extends to the climate system. It includes mineral crystals, microbial genes, animal behavior, ocean currents, island geology, fisheries, economics, and law. Each field describes one part. The reef joins them into one functioning structure. For now, the essential processes can be stated quietly. Corals capture food and host photosynthetic symbionts. They raise the pH of a microscopic calcifying space and deposit aragonite beneath their tissues. Colonies reproduce, compete, fuse, fragment, and die. Other organisms add carbonate, remove it, cement it, bore through it, and convert it into sediment. Waves arrange the material into crests, slopes, lagoons, islands, and deep foundations. Energy enters through sunlight and food. Nutrients circulate through animals, algae, microbes, water, and sediment. Disturbance removes living cover. Recovery depends on surviving colonies, larvae, stable surfaces, grazing, and time. Human activity can weaken or support every one of these processes. The future reef will be shaped by both biology and decisions made far from the ocean. Power generation influences seawater temperature. Agriculture influences coastal nutrients. Urban planning influences sediment and sewage. Fishing policy influences food webs. Shipping influences physical damage and invasive species. Scientific funding influences what can be detected and restored. Community rights influence whether management is accepted. Coral conservation is therefore not only the work of marine biologists. It involves engineers, fishers, farmers, teachers, economists, governments, tourism operators, coastal residents, and energy systems. The reef itself does not distinguish among these sectors. It responds to the total conditions that reach it. A reduction in one pressure can produce measurable benefit. Clearer water increases light and reduces sediment cleaning. Higher herbivore abundance reduces algal competition. Stable rubble allows settlement. Lower heat exposure reduces bleaching. Greater genetic diversity increases the range of possible responses. Benefits accumulate just as stresses accumulate. This is the practical basis for cautious optimism. Reefs are damaged, but they are not passive. Living colonies continue to grow. Larvae continue to disperse. Populations continue to contain variation. Communities continue to protect and restore local waters. Scientific methods continue to improve. The remaining uncertainty concerns how much time and environmental stability these processes will receive. The answer depends largely on the speed of global and local action. There is no need to hold all of these details in memory tonight. The reef can remain simply as a system of small animals, mineral structures, moving water, and countless connected lives. Its science is complex, but its processes continue one slow increment at a time. May the steady movement of this long subject now fade quietly into the background. Good night, and rest well.